Are you ready, Camden? Good afternoon, everyone. So, welcome everyone to the 4th Sharing Science Symposium. I'd like to thank you all for coming. This is a very unique event. It's probably it's certainly one of my favorite events of the year. This symposium came about as a way to pay back the community, keep the community informed and engaged with the research around HIV that goes on in Houston. It's been going on for a long time, and we realize that the community supports our research, but unless you're going to open up the scientific literature and read the results of a paper of a study that was done here, you're never going to know what happened. If you volunteered for the project, if you helped enroll people, recruit people for the project, even if you never heard of the project, this is research that's happening in our community, and the community needs to hear the results of it and be engaged in discussions around it. So that's the idea of this Sharing Science Symposium. I'm Tom Giordano. I'm a faculty member at Baylor College of Medicine. My clinical work is in HIV. I'm the medical director at Thomas Street, which is the HIV clinic that Harris Health runs. And Camden Hallmark here from the city of Houston and I have kind of been running this thing for the last 4 years. This is our 4th Sharing Science Symposium. To my left is Jeffrey Campbell, who's with the city of Houston. He's going to co-moderate this symposium with me. And without further ado, I think we should just jump right in. So the first thing on the agenda is announcements. Camden.
Hi, good afternoon. So my name is Camden Hallmark. I'm an analyst with the Houston Health Department. The first thing I'm going to do is actually introduce, unfortunately, Marlene McNeese was not able to be here today. She is our assistant director, and she's over disease prevention and control at the City Health Department. So I'm going to play a short welcome from her.
Good afternoon. My name is Marlene McNeese. I'm the Acting Assistant Director for Disease Prevention and Control at the Houston Health Department. And I would like to welcome you today to our 4th convening of the Sharing Science Symposium, From Research to the Real World. I'd also like to thank our co-sponsor, Baylor College of Medicine, in particular, Dr. Thomas Giordano, who each and every year has helped us to bring this research information directly to you. In the community, there are many things that you can do to help support our work. We in the government alone cannot do it. We cannot rely exclusively on our academic partners. We need individuals in the community just like you to do your part. What you will learn today are many new things in research and HIV program implementation. I encourage you to ask questions and to engage. There are so many members around the audience today who are there to help you. I wish I could be there with you today, but I'd like to especially thank some of the members of the health department team who are helping to pull this together, and in particular, Mr. Camden Hallmark. Without his diligence to content and ensuring that all the logistics of the event are taken care of, this event would not be possible. And I would also encourage you to interact with him, Dr. Giordano, and other members of the research team today to ask questions and to really engage in thoughtful discussion about how you can take the information that you learn back to your circles of influence. Your people around you, your friends and your family, anyone who you know could benefit from this information. So I appreciate you coming. I welcome you again and enjoy your day today.
I promise I didn't pay her to say that. Promise. So what I'm going to do is ask that Raven Bradley, she's one of the planning members that helped pull all this together, come up, and she's going to tell us a little more about how the rest of the afternoon is going to work and answer anything you might probably have as question-wise.
Good afternoon, everyone. Welcome. I'm happy to have such a great turnout this year. And today, based on feedback from previous symposium, today we'll have an opportunity for networking. This way you'll get a chance to speak with the presenters, other community members, health department staff, and other organizations in HIV prevention and care. Some logistical housekeeping items. Sign in, please. If you're in the room, make sure you had a chance to stop by registration and sign in today. Any needs, please ask one of the volunteers. You can identify them with the purple ribbons. Restroom locations outside the door to your right, and parking concerns. If there's any questions regarding parking, please refer to the registration staff out front. So when you entered today, you saw bags sitting on your seat. In those bags, you have a blue folder. That folder contains the program and agenda for today, one pager on research, resources for PrEP, linkage to HIV medical care, and transgender resources for both providers and clients, as well as an evaluation. That's the front of your left side of your folder. Please make sure you fill out the evaluation. We really do take your feedback seriously, and we hope to improve the symposium moving forward each year using your feedback. You can leave it on the table, or you can leave it with registration staff as you leave today's event. Also, for any certificates of attendance, please find Camden Hallmark after the event, and you can also find his contact information on the resource page in your agenda booklet. Also for today, we have partners in the lobby. So they'll be located outside the door to your left across from registration. First, we have the Houston AIDS Research Team, who has individuals enrolled here in Houston in nearly 50 different HIV research trials. Their Community Advisory Board provides community input and is looking for new members. Please stop by and see Morena Kay outside at the table for more information. Also, we have the O Project. The O Project documents the response to HIV/AIDS in Houston, Harris County, and Southeast Texas through recording oral histories and making them available to the public. You can find that information and that partner outside at the table. Her name is Sarah, and she can provide you more information there. Lastly, questions. We really made sure we gave you lots of time to ask questions and for presenters to provide answers. So at the end of each presentation, you'll be able to ask questions, as well as the end of each segment, the panel will be able to provide answers to any questions you have. So we'll have staff in the room who will have microphones to bring them to you and allow you to ask questions. Also, this year, we're going to do something different, which is Slido. It's a website that will allow you to submit your questions electronically if you're not comfortable answering them out loud. It's just 1, 2, 3 steps, and you can submit your questions. And lastly, if those two options don't work, you can write your questions down on the note cards in your agenda booklet, and you can put them in the baskets here in the middle of the room on the countertops. Other than that, thank you all for joining us today, and hope you enjoy today's event.
Go ahead, Jeffrey.
Good afternoon, everyone. I'm Jeffrey Campbell, and I'm the HIV Program Manager for the Houston Health Department. Want to take this opportunity to thank you all one more time for coming out on this afternoon. Looks like, according to Camden, that each year that this event continues to grow, and I think we're at record numbers again this year, not just for registration, but for your actual participation in this room. We are about almost 40 years into this epidemic, and over the 40 years, we've seen so many different things happen. We've got so many national days, from World AIDS Day to National Latino AIDS Awareness Day to National Condom Day. We've got task forces here in Houston that span most of the populations and communities that are impacted highly by HIV and AIDS. And back in 1999, then Mayor Lee P. Brown declared a state of emergency among African Americans here in Houston in regards to new infections in HIV. But the one thing that I think that has brought us to where we are today, where we can say these, we can put these three letters, we can put an E with an N with a D, and we can say end, stating the reality that we have what it takes now, everything that we need to end new infections, not just in Houston, but all over the world. And that is really brought to us by the work that scientists and researchers are doing. Many do it behind the scenes. They never get to a mic. They never get to do a presentation. But to get us to this place where we can begin to, in many ways, exhale and say, I can see the end of this epidemic. 40 years ago, 36 years ago, that was unknown of and unheard of. And probably to say that many years ago, someone would have declared you insane. So we're excited on behalf of the health department to join in with Baylor College of Medicine and bring to you Sharing Science, this symposium. And I want to personally thank, because I've seen the work that they've been doing over the last few weeks, Camden and his team in putting what you see together for not just us at the health department and not just academicians, but individuals in our community. So thank you and have a good afternoon.
Okay, before I bring up the first speaker, just a couple minor, or a couple other comments. First, I want to thank, I know not all the speakers are here, but I really want to thank them as a group for coming, agreeing to speak at this, and taking the time to put this together. We're very, as researchers, we're all very used to giving talks to colleagues. It's another it's a kind of different task to give a talk for an audience that's not scientific researchers. And it's not, it just takes a different kind of language, right? Because you don't know the lingo and the jargon, and there's nothing wrong with that, but that's what we're trying to do here, is communicate those things. So I am charged with being an interpreter. If there's stuff that I detect, you guys, that may be going over people's heads because it's said in jargon or lingo that no one understands, or skipping critical points, it's not their fault. The presenters are tasked with giving this presentation in about 10 minutes and trying to cover a lot of ground. I'll interpret if necessary, but please, if you don't get something, you guys are the reason we're here, right? So if you don't get something, ask a question. There's, some of these things we think about all the time, but you may not have thought of in these terms. So please feel free to ask questions, whether it's in person or using one of the other methods available. There's nothing wrong with that. We want that to happen. This is all about engagement. That being said, we do have a lot of people to pack in here, so we're going to try to stick to the timeline. If I say we can't take any more questions, it's nothing personal. It's just the reality of the time, the agenda that we have. I think I cut Bruce off last year a couple times, right? So that, that happens. And then the last thing is the feedback is critical. Please complete your feedback forms. If you stay for half of it, just evaluate the ones that you saw before you leave. Give us that information, because we really do read every single comment, look at every rating, and want to adjust the conference for next year. Okay, that's enough of me. Let's get to the program. So the first speaker is actually Camden Hallmark. From the city of Houston. Because the city organized this thing, he's going to get 5 more minutes than every other speaker. Camden's going to give you updates on 2 projects, interventional surveillance, so Project Pride and Beyond.
So thank you very much. And part of the reason why I am taking those extra 5 minutes is because there's a little bit to cram into this presentation. As some of you know, we at the city have been working on a project called Project Pride for the last 3 years. So I'm going to talk a little bit about that project. I definitely can't cover all of it. But I want to give some background about what this project entailed and why we started this. So in 2015, we were looking at really 2 major advances in HIV prevention. The first was treatment as prevention. So this is really about the use of treatment in those who are already living with HIV, and that treatment can lower the virus in one's body to a level where it actually prevents transmission. So it's really preventing transmission to a level where it's virtually zero. The other major advance that we were looking into at this time was PrEP, or pre-exposure prophylaxis. So this is a daily pill that, if used by people who are HIV negative, it can actually prevent HIV by over 90%. And so one of the things we wanted to do is see what that looked like here in Houston. One of the challenges with this was, did we have data to look at this? So at that time, we were looking at 2014 data because it was 2015. And we wanted to see how many people living with HIV in the city actually had achieved viral suppression, or that level of virus in the body where hopefully there would be no more transmissions. And what we found is 55% of the people living with HIV appeared to be virally suppressed. So we weren't too happy with that number. And another thing we looked at the same time was, did we have any data on PrEP, or pre-exposure prophylaxis? Unfortunately, we did not. That is not something that is reportable to the health department. When folks are retained into PrEP care, that's not reportable to us. But what we did look at is how many people might be eligible for PrEP in the US. And the CDC put out a study at this point saying that they estimated 1 in 4, so sexually active men who have sex with men, or MSM, might be eligible for PrEP. We paid very close attention to that number because about 67% of our diagnoses at this point were MSM. So we looked there. We also looked at people who inject drugs. About 1 in 5 might be eligible in the US, and then 1 in 200 for heterosexually active adults. So another thing to note, I've already kind of started talking a little bit about data. The reason for this is we do receive quite a bit of data at the health department. This is reported by laboratories and also by providers. So why do we get this information? It's really about getting information around public health emergencies or trends in data. And this isn't limited to just HIV. So Zika, Ebola, measles, these are things that we're monitoring on an ongoing basis. It serves as an early warning system for public health. We also want to document how interventions are going. We don't want to just go out and do a lot of stuff without knowing how good we're doing. So we want to make sure that we're actually making an impact. It also helps to monitor and clarify where information should come to inform policy. So for example, let's say, so this is a pretty straightforward graph around new diagnoses. If we see that there are new diagnoses increasing, we might want to target efforts to increase HIV testing, for example. But one of the critical pieces of this project we were looking at is called data to care. So this is using laboratory data to identify people who may be out of medical care. So if you're a person living with HIV, you might be going in once a year, every 6 months ideally, and getting some kind of laboratory tests done. Those laboratory tests get reported to the health department. And this intervention really looks at seeing if someone is potentially out of care and helping to assist them get back into care or initially get them into care. The whole idea, so around interventional surveillance, is simply taking this data and creating, planning, and implementing some kind of prevention intervention. So what did we do? There was some funding announced from CDC around this time for both PrEP and data to care. We were successful in attaining that funding for a 3-year, $4.5 million a year grant from 2015 to 2018. And the focus aligned very well with where we were seeing new diagnoses in Houston, which is men who have sex with men, transgender persons, and persons of color. Unfortunately, I do want to note here, we were unable to directly pay for pre-exposure prophylaxis, or PrEP. We could not pay for the medication using CDC funding. But this funding was for increasing supply and demand. So we wanted to increase people asking for PrEP from their providers and making sure that the providers were equipped to actually provide PrEP. We wanted to make sure that efforts were coordinated in the city. So if one provider was doing a great job, making sure that they had an ability to share that with other providers. Also, any kind of training, protocols, policies to try to facilitate this linkage and referral to PrEP. So what did we do? Again, this was a massive project, but I do want to highlight some of the really larger points with providers that we did. We wanted to target at least 100 providers. The first year, we got materials up and running, we had toolkits put together, and staff on board. And by year 2 and 3, you can see we were reaching over 150 providers those years. So all in all, by the end of the project, we have about a month left, but we have reached over 300 providers, mostly through telephone calls and some drop-by visits. We also put together something that I think we're all really proud of at the health department, a PrEP provider advisory group, where providers that really want to share lessons learned have that ability. So we went from 12 providers to now 50 in year 3. So one of the other things we looked at is what were we doing around PrEP uptake? At this juncture, again, we really didn't have a good sense of how Houston was doing. One of the things that we did was we already had some HIV prevention contractors funded by the city that already provide HIV testing. Into their contracts, we integrated PrEP education, and we looked at how that information was getting out to the community. We did a really good job, as you can see, in partnership with our providers of getting that information out about PrEP education. Then they do screening and identify people as PrEP eligible. You can see there is a drop off there. Not everybody is PrEP eligible, but also this is definitely a learning process. And those that receive PrEP referral, you might not always know how many people actually end up making it to a PrEP provider. But we do hope by year 3, we have a little bit more data left. This is some preliminary data for year 3 that we see some increases. So I have to credit our PrEP coordinator. Many of you know Megan Cannon. She is our PrEP coordinator in Houston for the last 3 years, and she really put together a great summary of what some of the barriers and facilitators were of getting this information out. So one was definitely misunderstanding on definitions of screening, eligibility, and referral. We learned a lot in that process. And then a lot of the staff that are working in HIV right now, they have a really high level of comfort talking to people about their sexual risk and about PrEP, but they don't really have as much experience early on with PrEP around personal knowledge and personal experience with PrEP. So there was a bit of a disconnect there. Some systemic changes. One of the things that we had to learn throughout this process was about what we thought the testing reality was on the ground and then what it really was. So we did some focus groups with the PrEP providers and our prevention contractors and really revised a lot of our forms to collect better data and to understand their processes better and how it varies from agency to agency. Also, we had an inability to monitor PrEP linkages. That still exists to this day. So if somebody asks me how much PrEP is going on in Houston, I do not have a good answer for you because, again, that's not something that's reportable to the health department. More staff trainings. We continue on this road in our partnership with other providers that are doing a great job and try to get more staff trained. And then, of course, really refining the referrals to not just giving someone a referral, but really the end goal being that they accept it and then actually make it over to a provider. I'm going to switch gears just a little bit to the other part of the project. So we've presented a couple years on this data to care, or using data to identify people who are living with HIV but might be out of medical care. One of the things that we wanted to do is, since this is really using data, it's not really encountering a client and talking to a client and saying, hey, do you need any kind of linkage to care? It's more about using data. We wanted to evaluate how this compared to other mechanisms where we're actually interacting face-to-face with the client first. And what we found is that about 33% of these referrals that we get in from data, we actually look in other data systems and find that these people are actually not either eligible or they don't live here anymore. So we don't even get to the point of referring them to a worker because we're able to close out that case just administratively in the office. Of those that we go out and try to find, we only locate about 34%. And then once we locate them, about 33% are ineligible, and about 10% of those that are eligible actually request care. So not a very high percentage there. We also receive some referrals from providers also using their data systems, and when they refer those over, those are also kind of data initiated referrals. When we compare those that are data initiated versus this really, warm handoff of a staff member of our team or another team at the health department talking to someone and the person saying, yes, please link me to care, we find that the results are very different, right? So we see this at the bottom, that we are able to locate fewer, and of course that more are ineligible, and that only about 11% request medical linkage. So what are all these results mean and why this matters? For PrEP, there was a high interest in coordination, so we see that through the PrEP advisory group. Monitoring success continues to be difficult. This was not something that I would say got easier along the way. It remains a barrier for all the PrEP providers about linkage and retention to PrEP, and that not being something reportable. If two providers are working together, it's very difficult for one to confirm with the other that a PrEP appointment was attended. Some facilitators, an in-house PrEP champion really works at provider training, either from the health department and or from Gilead, who's the manufacturer of the one approved drug at this point for PrEP. and establishing protocols and forms that are really modeled after reality. This meant that we really needed to do a good job at talking to our providers and our partners. Some barriers, definitely resistance to change in larger organizations such as the health department, other large providers. This was definitely a challenge. Perceived patient cost. So this one was really interesting. Our prep coordinator, I got off the phone with her yesterday, and I think that her parting message around this was that a lot of patients are self-selecting themselves out of prep. They think that they can't afford it. They think that because they don't have insurance, that they can't access it. But a lot of times there are programs either at the provider or patient assistance programs that actually work for people to reduce this down to low cost or actually nothing. And then providers had some anticipated expectations of what their patients wanted that didn't actually end up being correct. They thought that maybe there would be low patient demand, and there really wasn't in some cases. So why does this matter? So several years into this, now CDC has reanalyzed some of this data and found that still that there's a small percentage of Americans who are actually accessing PrEP nationwide, and really that this can be increased. Data to care, a critical element for us that we learned is a new data system. So we had to adjust some of the things we were doing at the health department to make sure we could monitor success and prioritize things going forward. We found it's difficult to locate these clients that are initiated in this manner. Many are ineligible for services and few request medical linkage. However, the data I showed you in the first slide, that still exists. That's still a measure that all the health departments are answering to, which is that 55%, which hasn't made a lot of progress in the last couple of years. So what we found out is these numbers are not as accurate as we had hoped for, and really that CDC had hoped for, which is that there's a lot more contextual factors about who's truly out of care. We utilize existing data systems to conserve resources, but again, a lot of these folks might be living elsewhere, or there's some other factors going on, and not necessarily that there's as high of demand that we think. Beyond Project Pride. So a little CDC update. Data to care is now required as of 2018 for every health department that receives core CDC HIV prevention funding or surveillance funding. What does that mean in Houston? We got a really great head start. So a lot of other health departments are struggling right now, figuring out how they're going to do it. We've been doing this now for quite a while, so we have a good understanding of how we are going to prioritize this new mandate. Also, the health department was awarded another CDC demonstration project. It's very similar in that it uses data for action and really to detect increased levels of HIV transmission. The population for this is, population of focus is Hispanic and Latino, men who have sex with men, or MSM. And this will include several of those prevention strategies such as testing, linkage and reengagement, and PrEP. I would be remiss without saying thank you very much to our PI who could not be here today, Marlene McNeese, as well as a very extensive team that you'll see here that has worked on this project for many years now. And then we had a great pilot that also started with some collaborators. So thank you very much to all of these folks that made this possible.
Thanks, Camden. Sorry. We do have some time for questions right now for Camden. We've got about 5 minutes or so. There was a lot of material presented there, and if I may take a stab at summarizing it, there's not enough viral suppression in Houston, and the city was tried to tackle this in a couple different ways. Or there's too much HIV being transmitted or being still being passed in Houston. The city tried to tackle this in two different ways. One is increase the number of people who have viral suppression. They can't change that necessarily, but they can get people who are out of care and get them back to care. That's where data to care comes in. We have 82% viral suppression at Thomas Street. These are people who are coming to the clinic. The city as a whole has 55% because that includes people who aren't coming to medical care. So you've got to get people who are out of care back into the clinic. That's what they're trying to do with their databases. But what they found was it's very hard to do that. The yield is pretty low. And this is something that a lot of other places and earlier projects in Houston have shown. It's hard to do that. Doesn't mean it's not worth trying, but you can't expect it to be the end all and be all. And if you can make contact with people, it helps a lot. The second part of it was we got to increase PrEP, right? Because that's the defensive side of preventing HIV. And again, there's a lot of work to be done there, but they made some improvements in getting providers engaged and prescribing PrEP, and putting resources out there, and trying to figure out what the next steps are. So that's a kind of quick and dirty summary off the top of my head. So questions for Camden?
Why difficulties just to find Hispanic patients to get the patients and then get the treatment on PrEP?
So why is it difficult?
For the Hispanic patients.
So I think that there are some structural issues that go on for many of our populations, and that's definitely one population as well. And so there's been a lot of targeted outreach more in some of the upcoming projects. So one of the projects that I mentioned that we're really trying to work better to understand those structural barriers that might be occurring in the community. It might be that we need more emphasis on understanding from the community what works, what messages work. So for example, when we're rolling out social marketing campaigns, we don't want it just targeted to one population. We want to make sure we're encompassing feedback from every population, that those messages make sense and that they fit that population.
Is any measure that using for the Hispanic community just to reach those community?
So the new project that we have a coordinator who's actually meeting with community, and he started really ramping up around in May to meet with community. There's a community advisory board with some of our partner agencies that is they're working all together to get messages directly from community members.
Okay, thank you.
If I may add, part of this is our problem, right? We have, if the last time you went to a doctor without not being in an emergency room and they said, oh, that's going to be $150, you may not go to a doctor thinking I can get PrEP because it's too expensive. So, so part of it is our message. We have to take this out to our network and say, you can get PrEP for free. There's ways to do it.
And I'd just like to add to that. Last year, for our media marketing campaigns, we contracted out additional dollars to 3 of our HIV prevention service organizations. FLOSS, which is a predominantly Hispanic serving organization, was one of those recipients, and they did an entire campaign on PrEP and data to care uptake in Spanish. So that was billboards, television commercials, radio spots, door hangers and the like. So there are different things going on in the community that speak specifically to the Latino population, but we do know there is a lot more work to do.
That means there's no response rate according to the number of Hispanic responding or getting back to these programs that offer that.
Other questions?
Howard Turner, City of Houston Health Department. My question is anybody looking into pediatric AIDS cases? And I don't know if this is where this question should be directed this time, just AIDS cases in terms of pediatrics. And also, Camden, are you guys, I'm hoping that you guys are working with the Houston Medical Monitoring Project in terms of getting data processed, and could you elaborate on those two issues if you can?
Sure, I can at least answer the last part of your question, and Dr. Giordano might be able to answer more about AIDS cases. But for MMP, or Medical Monitoring Project, that is a project that's going on among people living with HIV that the health department works on, and we do partner with them. We get referrals, so they're interacting with clients face to face. So most of our staff, including our TB staff that work in the health department, when they encounter clients and they might find someone that's out of care, we do work with them as well to get those referrals over and make sure that they're linked up with our service linkage program. And those folks are non-medical case managers that work on linkage and relinkage.
Okay.
Regarding pediatric AIDS, that's fortunately a rare situation these days. I'm going to ask you maybe to hook up with Mary Paul, who is in the white jacket there. At the networking break, she knows more about that than anyone in the room. I do want to move on, I'm sorry, to the next speaker. Again, we will have time for questions as a group, and then there's the networking break, which is designed to allow one-on-one interaction. So the next speaker is Dr. Jordan Lake. Jordan is associate professor at the UT Houston School of Medicine, McGovern School of Medicine, and a provider at Thomas Street, and she is going to talk about understanding the needs and concerns of transgender women living with HIV.
Thanks, Tom. So hi, everybody, and I'd like to just thank the organizers for inviting me to speak about this topic today. It's one that I've been working on for a while as an HIV and gender care provider at Thomas Street, and one that's close to my heart. So why do we do this study? So both HIV medications and feminizing hormonal therapies, which we're going to abbreviate here as FHT, and when I say that, I mostly mean estrogens or testosterone blockers, are really important components of medical care for transgender women living with HIV. However, both HIV medications and hormones have side effects, and they have the potential to affect each other's efficacy, and that's what we call a drug-drug interaction. So drug-drug interactions can go multiple ways. We can think about the HIV medications decreasing the efficacy of the hormones. We can think about the hormones decreasing the efficacy of the HIV medications. You can think of both of those things happening at the same time, or you can think about a scenario where when you're adding those two types of medications together, you may experience side effects that you weren't previously experiencing if you had either one of those types of medications separately. And really no data assesses drug-drug interactions between HIV medications and the doses of hormones that are very commonly used among transgender women. There's some data about interactions between hormones at the levels used for contraception by cisgender women and HIV medications. The modern contraception tends to use low doses of hormones, and so we're talking 10, sometimes more than 10 times the amount of estrogen that a trans woman may be using. And I don't think it's appropriate to extrapolate from studies of oral contraception to really understand safety issues for trans women. And then there is really also very little data to talk about knowledge of and concerns about drug-drug interactions that trans women have. So we did a survey of adult trans women living with and without HIV. I will say potential participants for this study did have to identify as being a trans woman or trans feminine. And for those who were living with HIV, they needed to be on medications and have a suppressed viral load. And the reason for that is this survey was actually conducted in the context of a larger study that was occurring in Los Angeles and Houston that's trying to understand how HIV infection and hormones affect risk of heart disease and stroke. And that is best studied when somebody is suppressed on their medications, and so that's why we had the requirement of that. But for the data I'm going to present to you here, our primary focus was really understanding the knowledge of and concerns about interactions between HIV medications and hormones, and whether or not participants self-reported using their medications differently than prescribed due to concerns about interactions between their medications. And so this is our patient population. We're actually still enrolling. We have about 160 trans women enrolled now in the study. But here you can see for the first 87, the mean age is 45 years old. About half of those are living with HIV. The majority are persons of color. And for the HIV positive women, I can show you over here. So we already said everybody has to be on medications and have undetectable viral load, and you can see T cells are pretty high, over 500. So a generally healthy population from the HIV standpoint. So what did we learn from the survey? So starting out, of the 87 trans women who completed the survey, most, 64%, were using hormones currently. Of those who were not using hormones, 20% had plans to use them in the near future, and only 16% had no plans to use hormone therapy. And you can see that whether someone was living with HIV or not, the results were pretty similar in terms of the number of people using hormones, planning future use, or not planning any future use. And so 64% were using hormones, and only 70% had ever talked to their doctor about the potential side effects of hormones. Now, some of this is the fact that 25% of the trans women who are using hormones were getting their hormones off the street or from friends. So obviously, if you're getting your hormones outside of a supervised medical setting, you're not going to be having a conversation with your doctor about it, right? Or you might, but you might be hesitant to let the person know that you're getting hormones outside of the medical setting. So overall, we saw that people were not having the conversations as often as we'd hoped. And it was even worse, if you were not living with HIV, almost 80% of folks were talking to their doctors about hormone side effects. But for those living with HIV, only 60% were talking to their doctors about side effects. So that is one main point here. The second main point is that 57% said that they were worried that their hormones and their HIV medications were interacting. To me, if 60% of people are worried, roughly, that's, a huge deal. And although 57% were concerned that there were interactions, less than half had ever had a discussion about this with their provider. So again, some people are not talking to their doctors because they're getting medications outside of the system, but plenty were getting prescribed hormones and still not having these conversations with their doctors. And then of the subset of folks living with HIV, we asked just straight out, are you taking your hormones or your HIV medications differently than they're prescribed to you due to concerns of drug-drug interactions? And everybody's HIV medication is prescribed, so this allows people to ask the questions whether or not their hormones are prescribed to them. And 28% said that they were taking their HIV medications differently than what they were prescribed to them. 28% separately said they were taking their hormones differently than prescribed, and about 8 folks in this group, or about 15%, were saying they were taking both differently than prescribed. And they were really just freewheeling in terms of making decisions for themselves about when to take HIV medications versus when to take hormones. So what does this mean and why does it matter? Well, I think it's just important as a basic fact that the trans women in this study were frequently expressing concerns that their HIV medications and hormones were either combining to cause extra side effects or were having efficacy interactions, but they were not discussing these concerns for the most part with their healthcare providers, with less than half of people having had a discussion. And secondly, that these concerns were the exact reason why people said they were taking their medicines differently than prescribed. We gave people options for why you might take your medicines differently than prescribed, and 40% specifically said it was because they were worried that the drugs were interacting with each other. And of course, we know that suboptimal HIV adherence increases the risk of developing treatment failure. Nobody wants treatment failure. And then with intermittent hormone use, you may either have suboptimal efficacy, so you may not get all of the benefits of being on hormones, and or there's data to show that when you take hormones intermittently, you may have increased risk of side effects, including heart attack and stroke, especially if you're taking more hormones than are prescribed to you at the time that you are taking them to sort of make up for that window of not having had them. And so these, I think, are really important points in terms of optimizing care for our trans women living with HIV. And so future studies are designed to understand these drug-drug interactions specifically between HIV medications and hormones, and to investigate ways that we can reduce this risk. We don't want anybody who needs HIV medications or wants hormones to go without either. So how do we do it safely? And because the lack of data exists, really more data is needed before we can provide healthcare providers with clinical recommendations for modifying risk. And then lastly, we know that hormone adherence and access to hormones and gender-affirming services is associated with higher HIV medication adherence. And now we're learning that people are concerned about drug-drug interactions and whether or not they should take these in combination. So the big summary is that our data suggests a need for comprehensive care programs for trans women living with HIV, or all trans persons living with HIV really, including clinical integration of hormone and HIV care provision services. And I'll end there just acknowledging this is another situation where it took a village to accomplish this study, and I want to thank our participants as well as all of the staff that devoted their time to making this happen. So thank you.
Thanks, Jordan. We have some time for questions.
What were the clients saying they thought their interactions were?
Yeah.
It was mixed. There was definitely some concern that the HIV medications would make the hormones not work. You know, that you would be taking hormones and not get the feminizing effects that you wanted because the HIV medications would prevent that. That was a common theme. This one?
Is there any science proving that, or is this just going off of speculation? So is there any kind of like tests that have been done, like even in the early stages, that possibly say that HIV medication and hormones are, is there any science?
Sure. So we mentioned that the doses of hormones that people use for birth control, and those data suggest that for most HIV medications, there's no clinically significant interaction, but the way that's always defined is, does the hormone keep the HIV medicine from working? It's never the other direction. And again, that's at low doses. And so there's been a couple studies, 2 were presented at the AIDS conference this summer, and that were specifically in trans women, and again, only looking at efficacy of HIV medications, and they were using 2 to 4 milligrams of estrogen a day, which is a little bit better than the birth control studies, but still, a lot of folks need 8 or 10 milligrams of estrogen a day, and you saw that even then, with 2 or 4, the amount of HIV medication in the blood went down 30%. So what you don't know is if you double that, if you go from 4 milligrams of estrogen to 8 milligrams, does the level of HIV medication drop even more? That's a huge question, and that's a big safety concern. And then neither of those studies went the other direction, and neither of them assessed how well the participants thought the hormones were working.
We have a question over here.
Okay, with the study, so this was only for trans women that you only got the transgender information from. So, okay, with the efficacy of the using hormones and the HIV drugs, so did you actually break it down into different categories from those that do injections, like they take the shots, the pills, the patch? Did you do all 3 of those types of studies, or did you just do plain and simple just hormone replacement therapy in general for trans women?
The data that I showed you combined all the different forms. And there was a mixture, of a little bit of everything. No one in the study was on patches or sprays, but there was a mixture of pills and shots.
Okay.
And remember, she doesn't have data saying there is a big interaction, but the data say that people are using both hormones and antiretroviral therapy at the same time, which is fine, but are concerned about interactions, and we don't know enough about it to give them good advice, and they're not talking to us providers about it, probably because some of them are getting it not from the healthcare system, and they're afraid, frankly. So, which is unfortunate.
No, that's the question I was asking, because most trans women, as a trans woman I am, most of us are trying to figure out those that are on HIV medications, that are to those that are not on it, which one would be the most effective treatment while they're on HIV medications? Is it the shot better for if I'm on HIV medications, or is the pill better if I'm on HIV medication? Is the patch better? That way, we can actually know the specific data. We can put trans women on that are HIV positive. That way, the hormone replacement therapy can work better for those that are positive and versus those that are negative.
Yeah, I think unfortunately we don't know the answers to those questions.
Yeah, so there are studies that people are trying to figure that out, but that's a huge unanswered question. And, there are some data in folks without HIV as to which hormones have this best safety profile versus patch pill shots. There's some of that data in the general population, but there's no data specific to folks living with HIV. So that's the data that we need, and that's why we need people participating in research so we can figure it out and provide safe care.
All right.
Okay, so we're gonna move on. There'll be some time for questions later. I feel like the trying to keep the trains run on time. It's hard because we could sit and talk about these things and have a great conversation all day. But Alessandro Gattoni is the next speaker. So this is a little different. Most of the time we try to have speakers who are doing stuff with humans, but that's not always the most important research that's going on. So we have a couple presenters today, Jason Kamada and Alessandro, who are talking about things that aren't even in humans yet. Andy Rice went through this a few years ago. He's sitting in the back and presented data from his lab that's all in cells and tissues or cells and test tubes. So this Alessandro is a professor. It says associate professor, but he's actually a professor at the Houston Methodist Research Institute, and he does work on drug delivery systems. How do we get medicines into people safely? And one of the things that he's using is nanoparticles. So his talk is entitled "Refillable Nanofluidic Implants for HIV PrEP, Pre-Exposure Prophylaxis." Okay.
Well, thank you very much. I'm delighted to be here today and talk about what we're doing in my laboratory at the Houston Methodist Research Institute. So I'm going to give you a brief introduction before to jump into what we're doing specifically in the field of HIV. Every time that anyone takes a medication, the concentration of the drug inside of your body goes up, reaches a certain peak, and then tapers down over time. And so this is the time in which then you need to take another administration of the drug. So you can take it normally through a pill or through injections. You always have this sort of roller coaster. And this roller coaster, as you can understand, is not very optimal. Of course, it's very convenient for a number of diseases, but think about it in the context of chronic diseases or treatment that require a patient to take medication every single day or multiple times per day. So every time that you have this roller coaster, you may have risk of getting your drug in concentration inside of your body. They are above what it's really needed by your body. And you get into potentially side effects, adverse side effects that you don't want. Also, whenever the concentration of the drug diminishes, of course, you don't have the same efficacy that you may need. And that's why you keep administering drugs one after a time through pills or injections. This is really suboptimal. And so this has motivated my laboratory to find alternative solution to achieve and maintain a therapeutic range of your drug for extended period of time or for as long as needed. Of course, once you're administering drug through pill or injection, there is a problem that is very significant in the context of HIV, which is adherence to the treatment. So you need to hope that the patient maintain and keeps taking the medication when is needed at the right doses. But there may be alternative solution for which a patient doesn't need really to remember that anymore, and the medication is delivered to the patient sort of in an autonomous way. And this is what I'm talking about today. So there are various technology in the pharmaceutical world that they are already being developed for various different type of pathologies. And of course you have polymeric system. They contain the drugs and that they are inserted, for example, underneath the skin or in different part of the body. And they degrade over time. And while they're degrading, they release the drug that is contained within the polymer. So these are roads, for example. They're used in the clinic, for example, for birth control. So these are inserted under the skin and then degrade. You have other systems that are not fully degradable, but they're still based on a polymer that releases the therapeutics. They are injections. They are inserted underneath the skin or intramuscularly, and they form a sort of like a depot, like a linear area where the drug is contained, and over time it is released again into the body. Also, we have other type of system, more complex. Osmotic pumps is a very intriguing device that allows for fluid from the body to enter inside of a reservoir and exert a force that eject drugs that is contained within a drug reservoir. So the drug ultimately is expelled through an orifice inside of the body. So it's a very ingenious system. And in fact, matter of fact, in Tarsha, which is one of the large pharmaceutical company, has been supported by the Bill and Melinda Gates Foundation in developing an implant such as this one for HIV pre-exposure prophylaxis. Of course, you are familiar for other people that you may know with other systems such as external pumps that deliver the drugs, but they are costly, of course. They're associated with other side effects because you need to have a transcutaneous catheter that delivers the drug inside of the body. They're really not as efficient. So what we decided to do in my lab is to find an alternative solution that was simple, it was potentially acceptable by patients, and also, of course, in terms of acceptability, we paid attention to the cost of the technology itself. So we realized that if we created membranes, tiny little membranes, so you can imagine a membrane, it can be a sheet of paper, right? Like that can be a membrane. There's a lot of little pores. Instead of using paper, we use silicon. Silicon is one of the materials that are used for making computer chip. So instead of making computer chip, we use the same technique that you use for making computer chip, and we create tiny, tiny little channels across this silicon device, this silicon membrane. And these channels are so small that we are showing right here are in the order of nanometers. So what is a nanometer? A nanometer is approximately, let's say, 50,000 times smaller than the width of our hair. So it's really, really small. And this can be done with very high accuracy. So you can create all this membrane precisely with an exact number of these channels and an exact size of this channel over and over. So the important thing is that we realize if we use these channels so small with our silicon membrane, then we can achieve a very sustained and constant delivery of drug without using any sort of pumping system. So that means that your implant or any sort of drug delivery can be just a drug reservoir that mounts this membrane, and the drug will always come out of the implant at the same rate for a long period of time. And you don't need to use any sort of pumps, any sort of electronics mounted on the implant. So the implant is conceptually and practically very simple. So in fact, our implant, I like to present it in analogy to an hourglass. So our implant, it looks very much like somewhat an hourglass. The reservoir, in the case of the hourglass, contains the sand. In our case, contains the drug. Our nanochannel are corresponding to the little stricture in the hourglass. And you see the sand, of course, keeps coming out at the same rate until everything is gone from the reservoir. This is exactly what we're doing. Now, let's look at the stricture. So this is our membrane, the membrane that we fabricate. And you see the nanochannel is this, in this structure, a complex structure where we have microchannel, nanochannel. Ultimately, what it matters is the nanochannels. And it's a very sophisticated, obviously, fabrication process that allow us to make nanochannel of any size and any shape. Why do we care about the size and the shape of this channel? I hope I will be able to transmit to you with this presentation the message about the technology is simpler than what it may seem. Ultimately, every drug that you want to deliver in a constant fashion, for example, has different properties. Some drugs are smaller molecules, some drugs are larger molecules, some drugs have a charge, a positive charge, some other are negatively charged. And therefore, we realized by testing all the different type of therapeutics, different drugs, an algorithm, a chart for which, knowing what are the properties of the molecule that we want to deliver, we can pick the exact size of the channels of our membrane so that we can achieve this constant and sustained release of the drug. And then, of course, we develop the membrane, and then we had to develop the implant itself. So the implant is just a drug reservoir. As you can see over here, all of this is a drug reservoir that mounts our membrane, the membrane with the nanochannels. And the drug passes through the pores inside of the membrane and comes off of the implant through the outlets of the implant. So what we like to do is insert our devices underneath the skin. And of course, in our preclinical studies, we use different locations. In our prospective use in the clinic, we are thinking about the inner arm, under the skin, where the implant is somewhat protected from the outside, but at the same time is easy to access and allow for a quick insertion via minimal invasive type of surgical procedure that can actually occur in the doctor's office as opposed to go through a regular surgery. So one important thing that you can see is that we have made a multitude of different implants, so different size and different shape depending on the drug that we use and the type of application and duration of the treatment. So of course, I'm now focusing on HIV, and I don't think I need to kind of present to you very much about the statistic about HIV, but it's important to note that while in several parts of the world, the HIV new infection rates has been decreasing over the years, even if not to a degree for which we are actually happy. There are areas in the world, such as Eastern Europe and Central Asia, where actually the degree of new infection is being going up by 60%. So it still is a very alarming scenario still. Looking at HIV pre-exposure prophylaxis, we know from many different clinical trials that the drug developed by Gilead Sciences, Truvada, is in fact effective if taken regularly. And so I think the best way to summarize the results is the following. If you look at these numbers over here, from this clinical trial, IPREX clinical trial, you see that you have 100% efficacy. I mean, we're talking about 100% efficacy if the drug is taken 4 times per week. And then the efficacy is reduced to 84% if you, for example, don't take it 4 times a week and you go from 2 to 3 times. And then even lower than that, or you don't have any sort of significant prevention if you take it lower than 2 doses per week. So this is obviously very important. Now, the outcome results is that despite the fact that the drug is potentially 100% effective or very close to be 100% effective, the outcomes show that it's from the clinical trial, we are about 50% or a little bit less than 50% effective. And this is due to the poor adherence to the treatment. Even for individuals that are at risk of getting infected by HIV, there is poor adherence. And this is, of course, is a real problem. So now our implant comes handy, and we realized that we could offer our implant for HIV pre-exposure prophylaxis, and we applied to NIH funding. We received funding for developing an implant that could deliver HIV pre-exposure prophylaxis in patient for an extended period of time. And we are targeting now 6 months, so that we would have an implant that can be inserted under the skin, could deliver the drug for 6 months. And at the end of 6 months, instead of being removed and replaced with a new implant, we would simply reload the implant underneath the skin. So it would be a minimal type of invasive procedure just to regenerate the entire implant for it to be good to go again for other 6 months and so forth. And so the way we will reload it is using 2 ports that would be easily located underneath the skin. And we would enter with 2 needles through the skin and load the drug directly inside of the reservoir. So these are 2 of the implants that we have used in our preclinical studies. Of course, we were studying 2 different drugs, TAF and FTC, so tenofovir alafenamide fumarate, and tricytabine. Now, you may immediately appreciate the difference in size of these implants. And in fact, yes, some of this drug, tricytabine, requires a lot of drug per day. So it really needs to be big because you need to have enough drug inside of your implant that can contain for a serial amount of time. This other drug, TAF, requires much less per unit time. And so we developed both of them because this was part of our project that we received funding from the NIH. And we moved forward and we did preliminary studies in non-human primates where we inserted the implant underneath the skin. You see here an X-ray where both implants are showing. And then we were monitoring both the drug inside of the body, inside of the tissue, as well as in the peripheral blood mononucleocytes, which are the cells that you really care about and where you want to see the concentration. And we had 2 important results. The first important result that is for TAF, the tenofovir alafenamide fumarate, we were able to maintain preventive level and actually exceed the preventive level for 70 days. Now, 70 days is the time that we set for this study, so we interrupted potentially earlier. We probably could have continued for a longer period of time. Again, we are targeting now 6 months. But anyway, we were able to maintain and exceed preventive level for 70 days for TAF. For FTC, the other drug that required a lot more delivered, we were not able to achieve the preventive levels. So now we are working to increase, obviously, the release profile so that both drugs could be released at the level that it's actually needed. One thing that was important, we were able to demonstrate at day 70 we could reload the implant through this very simple procedure. You see the 2 needles penetrating through the skin. On one side, the drug is loaded. On the other side, what is contained inside of the implant is actually removed. and we're able to do this flawlessly. Another important thing, of course, you may criticize and say, okay, well, the implant there is showing pretty nicely, but what happened in a case of in humans, really, where the different thickness of the skin may play a role. You may have some challenges in locating the implants and the ports. And so we perform preliminary studies. We're using a skin ultrasound that is very readily available in many clinics, and that we were able to locate the two ports. They are in fact the two darker areas that you see in the image. So there was a very nice confirmation of the fact that regardless of the thickness of the skin, we could locate the ports and refill the implant in need. One of the important things that is part of our project is making sure that the drug that we're using is adequate for an implantable device. And in fact, we know that the drug that has been developed originally for an oral formulation for a pill is not stable, chemically stable for a long period of time. And so we are developing formulation of the same drug that maintains stability for extended period of time. And this graph is to show that while you see the stability, this graph over here show that the stability of the drug, the TAF alone decreases over time dramatically, while instead we were able to obtain a formulation that is now stable for a long period of time, which is an important step forward. So I talk about the drugs from Gilead Sciences. There are other drugs that are under study in clinical trial. Cabotegravir is another one that could be used potentially for prevention, and we are also working with that. Of course, in the case of cabotegravir, we had different challenges. That molecule is very hydrophobic. In other words, doesn't really like to stay in water. So it's difficult really to dissolve it. And so what we did, we had to create a little formulation using beta-cyclodextrin, which is another molecule that is used very much in the pharmaceutical world, and we were able to demonstrate preventive level and maintain preventive level in preclinical studies. So this is in the context of a 91-day study, so about 3 months. And we are expanding this further to extend it to, again, the time frame of 6 months. Now, I want to acknowledge the team that is working on this with us. Jagan Sastry from MD Anderson, Roberto Arduino from UT Health Science Center and Medical School, Mark Marzinki from Johns Hopkins, Peter Anderson from University of Colorado in Denver, Jason Kimata, who is sitting right here with us from Baylor College of Medicine, and Pramod Nehete and Catherine Shelton from MD Anderson in Bastrop. So it's a very large team of scientists all dedicated to this project. I want also to acknowledge the team, my laboratory that you see right here at the Houston Methodist Research Institute, and the funding sources. And of course, I think for those of you that are interested in knowing what are the updates in our research, we have our also laboratory page as well as our social media pages where we post all our achievements and results. So if any of you is interested, you can follow our new achievements online directly. Thank you.
Thanks, Alessandro. So I hope you got a flavor of the challenges. This is, you think, oh, just shoot it under the arm or put it in a little thing and it'll come out. This is very challenging research. It's taking a team and quite some time to get this done, but Alessandro's team is making great progress. He quickly mentioned that this was done in 2 primates. I don't know if that registered with everyone. So they're not in humans yet. These are done in what were you using there? The 2, was it macaques or?
Macaques.
2 macaques. So.
And this is correct. So one important conversation that we are currently having, despite the fact that we are still in preclinical studies, is the Bill and Melinda Gates Foundation is following our progresses and has a potential interest in supporting the clinical trial.
In a clinical, yeah.
So this is a good development.
Yeah. So we're now at the point in the agenda where we're panel questions. So feel free to ask questions to any of the 3 last speakers, Camden, Jordan, and Alessandro, or things that might be to more than one of them.
Hi, Dr. Grottoni. Thank you. That's amazing research. Do you think there's implications for HIV treatment as well?
Yes, absolutely. So we think we start with prevention. The reason why we started with prevention is also because the amount of drug that is needed, that is delivered, is less than what is needed for treatment. And so, of course, we try first in the preventive setting, but the treatment, of course, will be the most logic continuation of our study.
The question I want to ask you is, with the 2 injection needles to refill the medication and to take that, do you do that simultaneously? And how difficult is that?
Yes, so you saw the little procedure that we have done, of course, experimentally, but ultimately we envision providing a doctor that would do the procedure with a little device where the needle penetrates simultaneously. So imagine a little tool designed specifically for this task, where you just need to inject on one side and the other part of the other sort of needle comes off, and so collect all the content of the implant autonomously. So it's a simple technology that we are designing.
And by the way, I said I think I said these were non-human primates, and Alessandro said macaques. Macaques are monkeys, what people would call monkey. So that's what these have been done in, small monkeys.
Hello, my name is.
To be clear,
that was loud. My name is Raquel Hines. I'm with City Houston Health Department Epidemiology. I was curious about the cost of these devices. Is it cost effective compared to the current prevention medications that we have now?
Yeah, this is a great question, and thank you for that. I didn't mention, in fact, this, but I said, in fact, we are considering the cost aspect of this. So while developing the implant per se is a very simple device, the only technologically advanced component is the membrane. And the membrane is usually fabricated, as I mentioned, with the same technique that you use to make computer chip. So the same way that happens with computer chip, you can make them in large number in parallel, which makes a component that is very technologically advanced extremely cheap. And so, in fact, we can fabricate these devices 700 in the same round of fabrication, and that dramatically cuts down the cost. But to give you, obviously I cannot give you an exact number of how much that will cost, but I can certainly say that the overall implant, the cost of the overall implant will be primarily due to the cost of the drug and not of the implant and the membrane per se.
Nothing new about that.
I'm going back to the first speaker or whoever made the presentation, something dealing with when you were doing data, you mentioned something about Latino population, I don't know, men, women, whoever. I was just wondering, the data you collected, is it going to be a study on the other races or this and that? I'm just curious about the HIV findings. I'm just curious about what is the data, for instance, collected for instance, Houston, Texas. I'm just curious about, the different races. What is the data collected across the board? That's what I'm kind of curious. I just don't know if you're just making a start with just one group, and is it going to just go across the board, or just right now that's just the focus?
Sure, thank you. So actually the data we presented was not specific to Hispanic, Latino, MSM. That was a future project that we're working on that was funded by CDC that is Hispanic, Latino, men who have sex with men, or MSM. But the data that we first presented was among everyone. There was a focus on men who have sex with men, transgender persons, and people of color. But that wasn't specific just to Hispanic and Latino.
Hi, good afternoon. Beth Sheba Johnson, HIV Prevention Medical Scientist for Gilead Sciences. All the presentations have been wonderful, but this is for Camden. You had mentioned data, and so I don't know if the audience is aware of AIDSview. There is 3 zip data so that you can go online in this website or app and look for zip code data on PrEP utilization within the areas that you serve. So I just wanted to make people aware of that.
And thank you. And I also recommend that as well. And they've done some great graphics that show PrEP uptake over the years nationally. So that's a great source of data as well. So thank you for sharing that.
Hi, guys. Yes, great so far. My first question is actually related to the app. There are a number of questions that have been posted there. I wasn't sure when those are going to be answered. I posted some, and I see a number of them there already. One of them I wanted to ask was the the injections when it comes to refilling or refilling when it's already been implanted. I know when Fusion started, there were some injection side effects, redness, itching, that kind of stuff. I wasn't sure if there was anything similar that y'all have noticed within your research.
Well, not in the macaque study, not in the non-human primate studies. We have not observed a problem with that. Of course, those are typical side effects that you can have from any sort of injections, and so I suspect that would be similar in our case.
But probably not like Fusion, if I had to guess. Fusion,
I'm not familiar with
95% of injections led to injection site reactions with Fusion, so it's not going to be like that.
Right. I was referring to any general injection that you can have. I wasn't familiar with Fusion per se.
Hi. My question is for Camden. Camden, around the PrEP data utilization information that you shared, is there right now a consistent data dictionary for our PrEP collection? And I ask that because early in the day when we were talking about linkage to care, there was various conversations about what was defined as linkage to care, and that was on the care side until we all became consistent on what that definition was. And similarly, I find that there is inconsistency among the contractors as how we define education with PrEP, initiation with PrEP, linkage to PrEP, as prescriptions probably the easiest to define. But I was just curious whether we're going to get to a point where there is citywide a data dictionary for all of us to go by.
Thank you very much for that question. Actually, so our PrEP coordinator and I had a conversation about exactly that, which has been a learning experience for us and definitely learning from the experts, which are you all. And I think that has been a point of frustration and growing pains, is trying to figure that out. And what she left me with was that our continuum is pretty novel in that there's not a whole lot to look at nationally for guidelines around what that should look like. And I think instead of us waiting, right, for the government to act federally to figure this out, I don't know if that's quite on their list of priorities at the moment, is I think the PrEP advisory group is a great avenue to explore that. And I think together with you all and with us at the city, figuring out what's practical and accounting for all the different ways that prevention goes on the ground. So I think that's a really great avenue to explore, and I'm hoping we can work that out this year together. Thank you.
This is a question for Dr. Lake, dealing with the transgender study. Okay, did you also have a study on trans women that were on PrEP and on hormone replacement therapy?
So one person in that original study was on PrEP, and several others were about to initiate PrEP. And so there are folks, but that study was not specific to PrEP. Although because anybody who identified as a trans woman or trans feminine could enroll in that study, that was one of the things we were hoping to capture. But there's other studies going on, like HPTN 083, which we can talk about more later if you want, which is enrolling a large number of trans women, and it will look at specifically at some of those issues of PrEP plus hormones.
Okay, also, will you be doing also a study for trans men also?
So we would love to eventually grow and have a broader research profile. Right now, we have studies for trans women predominantly in our portfolio, but we've had lots of people asking for studies in trans men, and we hope to get funding to do some of those, and we know that there's a great need. Much less is known about trans men and HIV risk and or living with HIV. The hormone issue seems to be less with trans men just because of the way testosterone works in the body compared to estrogen, but there are other issues that certainly need to be explored, and hopefully we'll be doing that in the future.
All right.
I did want to ask if there's some electronic questions we can go to, Ricardo.
Yes. 2 seconds. Sorry.
Okay.
May I ask a question while he's getting to that?
Sure.
Okay. I just wanted to thank everyone. I haven't been able to hear all of the presentations due to being at the table, but one common theme that I noticed throughout was that there are a lot of concerns and misconceptions that individuals face, whether it's self-selecting themselves because of their concerns about PrEP, or their concerns about the drug-drug interactions with hormones and PrEP or ARVs, or concerns about injections because of other things that they've seen previously. And so I was wondering about, I guess, thoughts about the use of, tools such as social media and, peer educators in various components to try to combat some of the concerns that people have. Because, there are websites, I know that a lot of you are using social media, but in terms of people's internal conversations within their community forums and with, and so forth, how to, kind of get past that sub-layer of fears that people have that they may not necessarily share as openly with their providers or researchers.
It's a great point. That's part of the reason we're here, is to get some of these messages out to the community and hear from the community what the messages are that are out there in circulation. I don't know if social media is the right way when it gets to research level discussions, but it certainly is a powerful tool for trying to influence opinion. So, if there's someone out here who has 1,000 followers on Twitter and wants to tweet something about this meeting, go for it. I have about 5 followers on Twitter, so I wouldn't be very effective.
At least you have a Twitter.
I do have a Twitter. I haven't tweeted in many, many, many months. So, Veronica, you're spot on. I don't know.
I'm going to go to electronic question.
Yes, the first question from Slido is an easy one. Will these presentations be available?
Yes.
Okay, so let's go to a little more difficult one, which is, are you all ready?
Yeah, their past years are also available online.
It's going to be online, and the link is posted on the resources pages in your booklet.
If you don't have the booklet, then just Google it.
So the first question from Slido is, could the immigration status have an influence in the number of Hispanic and Latino population in PrEP?
Another easy question to answer, yes. Now, there are healthcare systems and healthcare organizations that will care for people regardless of their immigration status and can get medication for people regardless of their immigration status. That's not the issue. The issue is often at the community level or the individual level, people are afraid, and not unreasonably afraid to come in. But that's another area where the community messaging is critical because they won't necessarily believe me, but they might believe a peer in the community who says, you can go there, they're not going to, I can speak for Thomas Street, no one asks for papers. They just want to, as long as you can show you live here, you're good to go. And there's other financial issues and stuff like that, but no one's going to ask for papers.
Another question from Slido is there any recruitment information for the FHT and HIV study that we can share with the community?
Yes, there is. So Marenike in the back is going to be at our Houston AIDS research team table outside, and she has a number of flyers for the different studies going on. I'd be happy to talk to you about it. That's also available on our website, and the Google phone number that is on the cards is available 24/7.
And I do just want to give Jeffrey a moment. He was going to talk a little bit about what we're doing with social marketing to follow up to Marenike's question.
So the City of Houston, the project that Camden talked about, Project Pride, when we were awarded this particular funding from CDC, a big portion of that funding is related to media marketing around PrEP and data to care or treatment as prevention. And one of the things that we've been able to successfully do over, it took a long time, was to sign on a contractor, Gilbert Communication, that is now working with us. They started by conducting focus groups to evaluate the marketing campaigns that were co-branded along with 3 of our local HIV prevention service providers, and now they're doing the work of actually creating a singularly branded Houston Health Department media marketing campaign that focuses both on PrEP and data to care. So a lot of that work is going to be centered around social marketing or utilizing social marketing, because a large portion of the target audiences that we're looking at to engage are young people who use social media marketing on a daily basis, and I would dare say on a 10-minute by 10-minute basis. So we will be launching that campaign starting, I'm going to say, about December. So look forward to that.
Any more there? Or
Yeah.
let's do one more question.
There's a ton of questions on here. So tell me when to stop. But the next one is, how will the activities changes developed through Project Pride continue after the demonstration project ends this year?
So thank you for that. So one of the first things that we're doing is that study that's continuing, or really a project, not a study, among Hispanic and Latino MSM. So we're taking a lot of the lessons learned from that project and Data to Care and in PrEP, and applying that to that next project. And we're still continuing to work with our PrEP provider advisory group. So I think we had some great ideas here today of next steps. So definitely we're continuing many of those facets of the work.
Another question is, for a complete idea on PrEP awareness and uptake, we need to know what happens after a referral is given.
Yeah.
What strategies have been discussed to accomplish this?
So I will say this and plead a little ignorance of not knowing everything that's been discussed in the PrEP provider advisory group, but I think that that's been the best conduit so far to make those conversations happen. We've invited any new interested providers who want to get more involved with PrEP to sit on that council. And I think that communication, it's going to be challenging because you're talking about multiple agencies, not just one agency talking to the health department, for example, but those agencies being able to talk to one another. We have a similar challenge, but not as great of a challenge around linkage to care, about agencies being able to confirm linkage with one another. But I think that mechanism of getting everyone in the room on the provider side and being able to establish a common goal, which is all confirming linkage to each other, I think that's going to be the best way to actually begin that conversation.
Okay, we used 5 minutes of our networking time to extend the discussion, but I don't want to cut any more into the networking time. If you have questions, so we're going to reconvene. We're going to break now and reconvene at 10 minutes before before 3. Those clocks on the wall are actually accurate, surprisingly. So you can use those. So at 10 to 3, we're going to break now for the next 20 minutes or so. If you have questions, please feel free to approach any of the speakers. We're going to stick around. The point of this is to engage in one-on-one discussions. Thank you all very much. I'd like to go ahead and get started again, folks, please. So, quick show of hands. How many people in the room here are physicians, nurses, other healthcare professionals? How many people work in the healthcare field in some way? Case manager, social worker, frontline staff? How many people are community not engaged in healthcare in a professional way? Volunteers, advocates? Cool, good, excellent. Thank you all for coming. All right, so without further ado, next speaker is Dr. Mary Paul. Mary is associate professor at Baylor College of Medicine, and she is a pediatrician, adolescent provider. So her topic today is research in youth.
Good afternoon. I'd like to thank the organizers for inviting me here to speak with you. I'm going to touch on two projects. The first one is the Adolescent Trials Network, which deals with prevention and treatment for HIV-infected youth. And then the second is the Pediatric HIV/AIDS Cohort Study, which deals with perinatally infected youth. And so the first project is around PrEP. And there have been a lot of PrEP studies of Truvada, which is the 1 pill once a day, but not in youth. And so this is one of the first studies in youth. And why did we do this study? Well, if you look at new HIV diagnosis in the U.S. by age in 2016, you see that that age group of 20 to 29, you see a big number there that are being newly diagnosed. So we need to do better in preventing infection in young people, having options for young people so that they don't get infected. And so what we did was an HIV pre-exposure prophylaxis demonstration project and safety study for young MSM. Now there were two different studies, one for 18 to 22-year-olds and the other for 15 to 17-year-olds. They're all HIV negative. We were able to enroll on the 18 to 22-year-old study. When it came to the 15 to 17-year-old, the study required that the patient or the participant provide their own consent. And it took us 6 months to get that through the IRB. And because of that lag, the study group decided that we wouldn't be enrolling on this study. But we learned a lot, and Dr. Moore was involved with that and is going to talk about that next. They all had HIV, so both the studies were very similar. They all had HIV transmission risk behavior in the previous 6 months. They were given daily tenofovir, fumarate, emtricitabine, which is Truvada, and then they were followed over 48 weeks. Now, at the beginning, they were followed monthly for the first quarter, and then for the last 3 quarters, they were followed by quarter. It took, for each study, it took screening of over 2,000 to come up with the enrollees because they really had to have high-risk behavior. For the 18 to 22-year-old study, there were 200 participants, and the 15 to 17-year-old, 78 participants. And so what we're looking at here is a measure of adherence by looking at tenofovir levels in dried blood spots. And if you were here a few minutes ago, you heard that the desired level comes with about 4 or more pills per week. And so this is the 18 to 22-year-olds and the 15 to 17-year-olds. It's this top part of the bar in both of these graphs that show the good level, which is at least 4 pills a week. And you can see that over 50% had this level at week 4, week 8, week 12, so in that, when there was that close follow-up in that first quarter. And for the 15 to 17-year-olds, again, you hit about 50% with a good level here in the first quarter. And then it drops off dramatically when you change to following them quarterly and over time in the study. These are the publications of these data. And so what we found was over half in each group achieved protective drug levels. Adherence decreased with the quarterly visits, and the study did, the one for the younger youth, result in label indication for PrEP for youth. So 35 kilos, about 77 pounds or above. There were several infections on the study for both studies. So the rate for the older group was about 3 per 100,000 person years, and for the 15 to 17-year-old, it was double that, 6 per 100,000 person years. And it occurred, these infections occurred in people who weren't taking the PrEP, who had levels that were not detected or very, very low, and there was no resistance in the study. So what do our results mean? They mean that youth need additional support to maintain high adherence to PrEP. So now we have a new project called iTech. It's NIH funded, and it's looking at technology innovative projects. So we have a new study called P3 that's just opening this week, and this is sort of the look of it. It's an app that will go on a mobile phone, a smartphone, has videos, has the ability for the subjects to communicate with each other about their PrEP experiences, and has different med strategies and different messages. And so we'll be testing this as well as testing adherence and usability, likability, and that in young people just starting PrEP. Now I'm going to move to the pediatric HIV/AIDS cohort study, and I'm going to talk about a study called AMP UP. And these are youth who are 18 and it's up to about 36 years of age who were born with HIV, and they're very different. These are the sites around the country that are participating. So far there's 475 youth enrolled, and we'll be talking about a pregnancy study. So why we did this study, with combination antiretroviral therapy, increasing numbers of perinatally HIV-infected young women are reaching childbearing age and becoming pregnant, but they're different. Their experience is unique. They've had lifelong infection. They've been exposed to mono and dual therapy, the older ones, and have resistance. And there's a challenge of maintaining long-term adherence to ART. Transitioning to adulthood and adult care is very disruptive, and they're experiencing, many of them, depression, isolation, stigma, and parental loss for some. Also aging up perinatal, and this is from the AMP UP study, showing that we're seeing lots of resistance. So this was a study of 175 youth who had within their chart a genotype. There was 75% prevalence of resistance, and that's any ARV resistance. To any of three classes, it was about 18%. We're not seeing integrase resistance, but to the other three classes. So we looked at pregnancy rates and postpartum virologic control among the perinatally infected young women in AMP UP. And what we found, so this is modeling. All of the women who were included, and the pregnancies that were included, had at least 2 postpartum viral loads. There were a total of 140 pregnancies, and that represents 99 women. So some had more than one pregnancy included. Live births was about 75%, and abortions and stillbirths was about 25%. And what we find for those women with live births is that they had RNAs around 400 around the time of delivery or less, but that within 12 weeks their viral loads went up to above 1,000. And so they weren't able to maintain their control of their HIV. So we observed lower rates of pregnancy when we compared the HIV-infected women compared to exposed but uninfected women who are also followed in the study. A high percentage, 42%, did have at least one pregnancy, and we need to be increasing postpartum support with respect to managing their HIV infection. The end of pregnancy may be a good time to intervene to ensure adherence levels attained during pregnancy are maintained in the postpartum period. And so those are my remarks for today. I do want to acknowledge Bill Shearer, who was a founder of our study group. all of our nurses, coordinators, data managers, lab personnel, investigators, social service providers, and counselors. Texas Children's Hospital, Harris Health System, where we have clinics as well. Also, Legacy Community Health. They collaborated with us for the HIV negative men who have sex with men that we enrolled on the PrEP study. Of course, the community, all the different organizations that we interact with, and our study volunteers. So I'd like to see if there are questions.
Yeah, thanks, Mary. So just a couple quick clarifications in case there's some confusion about it. Mary started with saying they couldn't enroll in the youngest age group, but then presented data from it. That's because the study was national, and so she was presenting data from across the country. Some of the older youth were enrolled in the study from Houston, but they were unable to enroll anyone in the younger cohort from Houston because of the issues that Keonta Moore is going to talk about in a second. So the data were from the entire country for that first one, just in case people were confused about that. The second study, it's a very unique group of people who were in that study. Just to make sure people are clear on this, these are women having babies, but these women were themselves HIV infected when they were in utero. Generally, these are women who have lived their whole life with HIV. So their moms were positive. The mom got pregnant. The baby was born positive. The baby is now grown up, which is awesome, and having babies. But what they found was that when this woman who's lived with HIV for her whole life, when she was pregnant, she took good care of herself. Her viral load was really low, and then after delivery, on average, the viral loads went up. Whether that's because they're too busy with the new baby, what's going on, is not clear from the data, but that's basically what they found. So we have a couple minutes for questions for Dr. Paul.
Mm-hmm.
Like postpartum depression?
Yeah, that's a good point, and that is definitely something that we're going to look into, the factors that resulted in the viremia postpartum, but it is something the pediatrician screens for, and we are taking part as well in something called centering parenting and screening for postpartum depression when we do the follow-up of women who have babies and do the testing of the babies, and it's not uncommon.
In the back corner there.
Of participants who had continuous or steady access to healthcare in terms of personal or private insurance or insurance through an employer versus those who were potentially receiving care through Medicaid coverage during their pregnancies?
For the women on the second study?
Yeah.
No, I think that my impression is that the women who are in care really during their pregnancy are very adherent to care and to medications, and it doesn't depend on who their payer source is, but that afterwards, that's when things become more difficult.
That's I think what I was just questioning, because if they had continued access to reliable medical care versus the cutoff when they stop receiving Medicaid coverage after their pregnancy, I was just wondering if that had any impact or was correlated with the results you found, because it seems pretty steep.
I mean, I don't know across other states how unique Texas is in only allowing a few months of Medicaid coverage after delivery. And remember, this is a national study, so the idiosyncrasies of Texas may not apply to the rest of the cohort. It's a great question, but I don't know that this study is the place to answer that. One more question.
Were any of the participants part of the Centering Pregnancy program, and did that impact postpartum, adherence to medication?
I'm sure that Dr. Levison is looking at that. This is a separate, some of them, yes, would have been in centering pregnancy. But we haven't looked specifically at that group as a separate project.
All right, thank you, Dr. Paul. So we're going to move on to Dr. Keonta Moore. Thanks. So Dr. Moore is a child health policy fellow at Rice in the Baker Institute for Public Policy and adjunct at Baylor and a pediatrician. And she did a study that Mary alluded to on legal barriers to adolescent participation in research about HIV.
Great. Thank you. Thank you so much. So I'm going to talk to you a little bit more in depth about what happened with Dr. Paul's study and the implications of that for anyone who's interested in doing research in adolescents where it's involving a clinical drug or the FDA. So why we did this study? Well, at the time, I ran Baylor's Research Ethics Consult Service, which no longer exists. But Mary, Dr. Paul, reached out to our service at the time when she was trying to get IRB approval for the ATN study that she just described for you in the adolescents that were 15 to 17, so they were legally minors, but they were at-risk minors, so they were young men that had sex with other men. And our IRB really interpreted both the FDA regulations, which the FDA basically says that any clinical trial, so any study involving an investigational drug, if it involves a minor or a child, you have to have parental permission. Well, the ATN study that Dr. Paul was doing explicitly said not to get parental permission. As many of you likely probably understand and know, this group is very at risk. There were challenges, worried about selection bias, that if you require parental permission, only those adolescents who had great relations with their parents or sort of were not the norm or the most at-risk population would be those enrolling. And so for that and a variety of other reasons, they did not want to have to get parental permission, but now you have this situation where the FDA is saying you have to have parental permission if you do clinical trials or research with a child. So the issue why we did this was the IRB did not approve Dr. Paul's study and ended up not improving it until after, and I'll describe later our results, we wrote a legal and ethical analysis for the IRB board. They eventually sort of understood the parameters and what was possible. They approved it, but as Dr. Paul described, it was too late. And so the question that we had is it ethical and legal for at-risk minors to self-consent, or in other words, to participate in research without parental permission in HIV prevention clinical trials? And so what did we do? Well, we did an ethical and legal analysis. So I'll go a little bit more into our ethical analysis, but for the legal analysis, basically, I looked at statutory history. We reviewed the statutes regarding sexually transmitted infections and whether or not minors could self-consent for medical treatment, for just STIs. We looked at whether or not states had HIV-specific statutes where minors were allowed to self-consent, and then we looked at the language of those statutes regarding whether or not any of them said that minors could self-consent to prevention, which is really what this was. And so we were in an interesting situation in that this was a drug that was given, right? So it was part of the FDA clinical trials, but the drug was given for prevention, which, you know, traditionally in medicine, that's something that's new and that our state laws really haven't caught up with. Now, we have laws regarding vaccinations, but the law thinks about that vaccinations or treats that differently than medication. So we found ourselves in a situation where really the law was very ambiguous as to whether or not we could, you know, reasonably consider prevention to be within the scope of the definition of treatment within the statute. And then ethically, we looked at autonomy, both the rights of the individual and the rights of the individual, the adolescent. So for an example, you know, adolescents probably around the age of 14, so the literature has shown that cognitively they have the ability to make decisions on their own. So part of the reason why the FDA requires, you know, parental permission is because they want to make sure that, you know, minors aren't being convinced to participate in things that have a lot of risk to them. And so, and the law is assuming that when it defines it as a child, this child doesn't have the ability to autonomously make decisions for themselves, right? And our Supreme Court and a variety of other laws have said that parents should and do have the right to make decisions for their child. But there then comes a point ethically, right, where you have this tension between the rights of the parent to make a decision legally for their child and then the evolving autonomous decision-making capacity of that minor. So by the age of 15, minors should be able to reasonably weigh risk, you know, be able to anticipate future foreseeable consequences. They should be able to express a preference. They should be able to align their own values with whatever is being presented with them. So, you know, obviously this is not a blanket thing, right? Like there's definitely protections that the IRB can and should put in place to ensure that if there are minors who are self-consenting without their parents, that they exhibit these clear characteristics of having autonomous decision making. But in this instance, we felt like the minors that, you know, and, you know, Dr. Paul and her colleagues had a very extensive consent process. They had lots of protections in place. And so for, you know, my job was not, you know, was really to hone in on, you know, provided that, yes, there's protections in place and that, you know, these adolescents have autonomous decision making, is it ethical to allow them to self-consent while weighing the parent's right to make decisions for their child? And so what we came up with was what we decided was that, you know, basically, yes, at-risk minors do have the mental capacity to consent, and they should be allowed to consent to participation in HIV prevention clinical trials without parental permission. Now, we also put a caveat in there that said, of course, we respect parents, right? We respect their rights as parents to make decisions and to protect their children. So all efforts should be made possible to make sure that, you know, there's an opportunity for the minor to engage the parents in this process, that safeguards are put in place to make sure minors are not being taken advantage of, that the consent process really makes sure that it's informed and it's in depth so minors really understand what they're actually consenting to. But, you know, that being said, we felt like, you know, given that, you know, all of those safeguards are in place, it is ethical for minors to self-consent. Then we had to think about the law, right? So I was sort of doing this simultaneously, thinking about the ethics and thinking about the law. And so, you know, as many of you know, all 50 states say that minors can consent without parental permission to sexually transmitted infection, you know, treatment for sexually transmitted infections. 34 states at the time of the study, so that's a caveat. I haven't done a legal review lately. say that, okay, it's okay for minors to consent to both testing and treatment, and that includes Texas. So our real issue here in Texas, it came down to, does the Texas statute and language of treatment include prevention? And we believe that it did. So after looking at, case law, looking at, we even looked at legislative history, what, the legislators were thinking about and their purpose behind the statute. We looked at the administrative codes that regulate, the treatment of medicine. We looked at insurance codes, which regulate and govern billing, right? There's many instances in medicine where, physicians or clinician will see a patient and bill for that visit, but it's all prevention. But our statutes don't really say anything about prevention, right? But we've been conducting business in our state in the healthcare field as if treatment included prevention. So for a variety of reasons, we felt that it does, and that access to PrEP and other sexually transmitted infection prevention services is not explicitly prohibited by Texas state law. We also felt that this reliance on state law was creating quite a bit of confusion. So in this instance, it was a national study, and while many people might say, and you might even say, well, they ended up doing the study, so what's the big deal? Like, why does it matter that we couldn't do it here in Texas? Well, you know that, many, if you've been around, right, you've heard social determinants of health, right? There's these environmental factors, these geographical place-based factors that influence health outcomes, health behaviors, and so to exclude an entire state from a study is potentially harmful for our population because there may be place-based influences on treatment adherence, on outcomes that were not considered in this national study. And so this is important for us to think about. And then lastly, we just made a recommendation that IRBs in Texas should really adopt the policy to include this broad, definition of treatment to include prevention. So I'd just like to thank my colleagues, Dr. Mary Paul and Dr. Amy McGuire, the Center for Medical Ethics and Health Policy. So questions? Sir?
So thank you for that research and getting that done and having some facts so folks can help make some decisions. But you mentioned that the IRBs can adopt policy, and that was a recommendation that was made?
IRBs, that was our recommendation.
Internal Review
and
Boards,
the
right.
Baylor, right, I don't know if they adopted fully, but they came around.
That's what I want to hear.
To
Recommendations are one thing, but I'd
Yes.
like to know whether or not they were adopted.
Yes.
And if they were adopted, can this be a precedent to be shared with other research universities and outlets so that they can also review the data to help improve research for anyone living with HIV, whatever age,
Yeah,
particularly the
why
youth?
It was published as well, so
Yes.
it's out there in the published literature.
Mhm.
Baylor approved it.
Thank you. This is Ruth Boozy, and I am very familiar with those issues because I do research with adolescents, so I know exactly the challenges are. So I am wondering, if you take, that process of understanding the interpretation of what adolescents, are allowed to do within, the law, what are your thoughts about the PrEP that's now available for adolescents? Can we get adolescents to use PrEP without parental consent?
In clinical, not in research.
Not in
just
research,
clinic.
right. But it's still like in the context of prevention, right? Prevention of high-risk individuals. So how can we apply what you have learned through your analysis to understanding whether or not we'll be able to get adolescents on PrEP without parental consent?
So the problem with that is getting the PrEP and the laboratory monitoring for the adolescent. So they may be on their parent's insurance, and the parent will find out in that way. Gilead, even though there's an indication, I haven't heard that they're allowing their programs where they provide medications are allowing minors to access that without parental consent. And then also, yeah, it's a matter of adherence as well. So if they're in the household, having them be adherent to their medication, even though they're trying to have it secretly with the parent in the household. So all of those issues. So we haven't begun prescribing to minors, but those will be the issues that we would be discussing with a minor who was trying to get PrEP without parental consent.
If a minor came in with their parent, then there's no problem.
No problem.
Mhm.
Yeah.
Yeah.
No problem.
But it's when they're trying to.
But to your point, right, we've laid the groundwork that yes, you can prescribe, right, off-label, or you said it's even now on-label, right? It's approved now,
It's on
right,
label.
for minors. But now you have a host of other ethical challenges around confidentiality and privacy of the minor, whether or not there's going to be retribution against the minor, if the parents find out. You have a host of other challenges that we did not address in this work.
Prescribe adolescents birth control without parental consent, certain funding streams. But if you're on Medicaid as an adolescent, we can prescribe you birth control without parental consent. So if a minor is on Medicaid, are we going to be able to use the same logic we use for the birth control? And even on birth control, we have the same issues of adherence, of secrecy, kind of keeping it away from parents. We have the same issues. So how are we going to be able to kind of really navigate that system?
So the state laws don't necessarily, all the state laws do not necessarily protect birth control or contraception, right? So you have to understand the laws that regulate the practice of medicine are distinct and different, even if you could apply the same ethical principles, right? So in Texas, we explicitly say in Texas that a minor can access HIV testing and treatment. That is a statutory law. So by law, minors have the legal right to access treatment and testing for HIV. For contraception, there's not an analogous law in Texas that says minors have the legal right to access birth control or contraception without parental consent. So you have to think within the legal framework before then you can apply the applicable ethical principles.
Okay, we could go on for
Yeah.
a long time on this topic, I'm sure. It's fascinating. Thank you both for great presentations. Okay, now, as Monty Python says, for something completely different, Jason Kamada. Jason is associate professor at Baylor, and he's going to talk about things that are not in humans, one of those presentations. So Jason's talk is eradication, still a fascinating topic, so pay attention. This is going to be a challenge. I'm warning you. I had trouble. I reviewed his slides 3 times before I could write the summary. So, eradication of HIV by T-cell immunotherapy. Jason.
You're scaring me, Tom. I'd first like to thank the organizers for inviting me.
Sure,
and
say
giving me
that
the
now.
Opportunity to speak to you and try and convey what we're doing. So I think everybody knows that there's been one individual cured of HIV infection, Timothy Brown, or the Berlin patient. And this has really, I think, stimulated and gotten, at least from the basic science side of things, to address the question of can HIV be cured? And I think, we all know that this is a real challenge, and mainly because antiretroviral therapy inhibits replication of the virus. As you know, it's quite effective at driving down the amount of virus in the body, but it doesn't cure infection. And perhaps the main reason for this is during antiretroviral therapy, the virus, which is infecting CD4 T cells in the body and destroying those cells, but during antiretroviral therapy, it drives down the virus, prevents it from replicating, But it doesn't get rid of the virus in these cells because the virus eventually goes into this silent state where it's not reproducing itself, and it's just sitting in these cells without dying off. Okay. The alternative is it's also found in protected sites within the body, protected as in sites where antiretroviral drugs are inaccessible, so the virus continues to replicate at this low level, but is being passed from one cell to another. And then also you have this effect where whether it's being silent or through mutation of the virus, it's able to escape from both the host immune response and unfortunately from drug suppression as well. So how can we help improve the immune response against HIV? Okay, so one of the major problems obviously is that it's infecting a very important cell of the host immune response, the CD4 T cell. So this compromises the ability of the immune system to work and clear the virus. So we decided that one thing we could do is tap into our colleagues at the Cell and Gene Therapy Center and revisit this issue of can we take and isolate HIV killer T cells, expand them outside the body, and reintroduce them so that they help control the infection. Now, I say revisit this issue because this has been tried once before, about 20 years ago, and it failed miserably. In part because the technology for doing this wasn't very good at the time. And currently, many of you may know from the news, doing this, which is referred to as T-cell immunotherapy, has become a very effective treatment for cancer and for treating other virus infections associated with cancer. But it hasn't been revisited with HIV. And one of the main factors that may be preventing its utility is that HIV targets CD4 T cells and kills them off. And we have to, if we're going to try this again, we have to come up with a way to protect those CD4 T cells from getting infected. Okay, so what did we do here? So first off, so by training, I'm a basic scientist. We study interactions of the virus with the cell. And so we use what we've been learning over the years to develop a new type of inhibitor. It is a cell-associated inhibitor that can protect CD4 T cells from getting infected. We had to revisit the idea of what the best way to take T cells from an individual, take them out of the body, stimulate them with proteins of the virus so that you could isolate these HIV-specific killer T cells. We then took the inhibitor and introduced it into these cells by genetic modification. And then we examined how well they would be able to kill HIV-infected cells. And all of this so far has only been done in the laboratory, as Tom had mentioned. So first, what is this inhibitor? How does it work? So the inhibitor we call X5AR for X5 artificial receptor. And what it does is, again, we genetically modify T cells to express it, CD4 expressing T cells. It gets displayed on the surface of these cells. And what it does is when the incoming HIV is going to infect the cell, it prevents it from engaging the molecule CD4, which it uses to enter the cell, and blocks its entry so that it can't replicate. And it works extremely effectively. In fact, these are all based on native antibody molecules, which normally target the virus. But in this context, they work quite a bit more effectively than the native antibody. And then we discovered something rather remarkable. which is that these cells expressing this cell-bound inhibitor on its surface, if you put it into a culture or a test tube of cells that have some infected cells and some uninfected cells, and look to see how well it's preventing replication of the virus, the cells are not only protected themselves, the cells that are expressing the molecule are protected from getting infected, but they're also capable of protecting the uninfected cells from getting infected. And it turns out that part of the reason why this is happening is because these molecules that are expressed on the surface of the cell are getting transferred over to the uninfected cells and protecting them, creating this protective barrier so that these infected cells are unable to transmit the virus over to the uninfected cells. Okay. The other thing they're doing is, in this context, is that they're capable of binding up the virus and internalizing them, and it appears that they degrade the virus so that there's no longer infectious virus there. So this was quite exciting for us, but we had to test the next step, which is, would they improve the killer activity of HIV-specific killer T cells? So going back to the original issue, which is that the virus is in the silent state during antiretroviral therapy, we had to be able to awaken the virus so that the virally infected cells can actually be seen by the HIV-specific killer T cells. And the way we do this is we add a compound that can induce these cells to start making virus again. And of course, this is all in the context of having antiretroviral therapy there, so it can't necessarily pass virus on to new uninfected cells. But this alone is not sufficient for allowing the killer cells that are modified with the X5AR to actually recognize these cells and dispose of them. And it's part because the virus, when it's being made, has a defense mechanism that prevents the killer cells from seeing it very well. So we figured out that if we add an anti-cancer drug that is already being tested in humans and used in humans for cancer therapy, it primes these cells or enables them to be primed to die. And when we add our HIV-specific killer T cells expressing our inhibitory molecule, it now effectively recognizes these cells, targets them to be killed off specifically while leaving the uninfected cells alone. Okay, so that's what we've determined so far. Again, this is all within the laboratory setting. So what does this mean? So it means that, first of all, we can overcome this resistance of HIV producing cells to the killer T cells by combining two things. One, an anti-cancer drug that prevents cells from surviving and primes the HIV producing cells to die. And then they can be cleared out or killed off using these HIV-specific killer T cells that have this specific molecule engineered to be expressed on them. Okay. And then I think it gives us some optimism that immune restoration with genetically modified HIV-specific killer T cells may in fact be a feasible way to eliminate infected cells, even perhaps in the absence of antiretroviral therapy. Okay, so I'm willing to take any questions. I just want to point out that this is in the collaboration of 3 laboratories. My own laboratory This is mainly the work of a graduate student, Anisha Misra, in collaboration with a graduate student and my colleague Paul Zhou at the Institute Pasteur of Shanghai, his students Michael Wen, and then another graduate student, Shabnam Patel, who works with Catherine Bollard, who used to be here at the Cell and Gene Therapy Center at Baylor College of Medicine, is now head of immunotherapy at the National Children's Hospital Research Institute in Washington, D.C. Thank you.
We have a question.
So the question on Slido is for the last presenter. It is not all adolescents meet this category as mentioned. Therefore, how do you determine the adolescents be included in the research?
Before we go to that, do we have questions for Jason? Just a couple to see if there's clarifying questions. People get it? It's confusing.
Okay. So this new immunotherapy, it works for people who are on each class of antiretrovirals and those off of antiretrovirals.
We're not at that point yet.
They're
This is just proof of concept in test tubes.
I know that he stimulated them from being hidden. Which antiretrovirals used to make the cell hidden is, I guess, my question.
Yes. So for those experiments that we did in the laboratory, the cells were taken from individuals on antiretroviral therapy. I don't know exactly which drugs they were using, but some contemporary art regimen.
Okay. Because I know that they changed some of the classes changed the membrane of the cell in a certain way. I'm just wondering if that could interact with, if different classes can interact with the attachment of that extra blocking channel.
So we haven't seen any issue with that.
Okay.
Yes.
And most of the drugs we use, 95% of the drugs that we use in people don't do that. The ones that do interact with the CD4 and CCR5 receptors are pretty rarely used. Probably less than 1% of people are on those drugs. So I doubt that comes into play in this particular research. It's a great question.
I can speak up. I was wondering, when you say bringing out the cells in the hidden space, or that are kept in hidden spaces, I'm assuming that means like brain, lymph nodes, where the antiretroviral therapies don't go, right?
Yes.
So what are the implications for that, like in the brain, doing that where it wouldn't be degenerative like it is now, sort of?
Yeah. So I'm assuming you're wondering, does immunotherapy or T cells pass into the brain? And yes, there are T cells in the CNS and brain. So I would think that it would be feasible for it to get there and control infection.
Well, the question I have is that the cells that go silent are the sleeper cells. When you say you awaken them in the test tube, when it's in the body, do you think it will have a reaction where it will awake all the cells at one time? Because I think that seems to be what the problem is that the body constantly produces, and some go to sleep and some stay awake. So my question is, would this awake all of them at one time enough to give you an opportunity to attack them at one time?
Or just, if I may, if they all awaken at the same time, could that alone be bad? Could you have a big immune reaction from that process?
Yeah.
Those are concerns.
Yeah. So those are legitimate concerns and a great question. And so actually it's been attempted to what they call the shock and kill method, because it was hoped that you could reawaken the virus that reawakens would kill off those cells, and while someone is still on antiretroviral therapy, it would prevent the infection from spreading. So you can see when some of these, what they call latency reversing agent, has been tried in a person, you can see a spike in the viral load, but you can't detect any measurable decrease in the amount of virus the person eventually has afterwards.
So we haven't been able to successfully
Yeah.
to shock and
Right.
really awaken all these cells at the same time.
Right.
No one's figured out how to do that yet in a person.
Right. So we hope that, if immunotherapy can be used, it will help with the kill part, right? And help clear out the reservoir of cells.
So let's open this up to questions for all the panelists now. You're certainly welcome to ask questions to Dr. Kamada still. But
I just had one more. I just was curious, like in conjunction to the immunotherapy, have y'all tried like a hydroxychloroquine, which is like an antimalarial, but to stimulate or modulate the immune system using hydroxychloroquine or something like similar to that?
So we've never tested that, but I believe there was a clinical trial to do that. I don't know what the results were.
does anyone?
Hi, it's Bathsheba again from Gilead. I want to go back to Dr. Moore and Dr. Paul regarding the Gilead's program for access for minors. It's the same as adults, and so going through their access program for the medication is the same, with the exception that you have to have an adult signature. So they have a form for that medication. It has to be signed by an adult, someone 18 and older. It can be the provider themselves, the prescriber. It could be a parental guardian or a legal guardian that can sign that. But it's the same otherwise to access that $7,200 a year.
So I just want to clarify. So there is a distinction between research and clinical practice.
Yeah.
So what we're talking about are the research challenges and what we found with adolescents participating in research. There's other regulations, that are even more vague that regulate the practice of medicine. And most of that is dictated regarding hospital policy, right? So Texas Children's has policies around confidentiality, around treatment of minors without, their parents. And so there's a distinction there between what we're talking about.
I totally understand that, but when the young lady mentioned
Yeah,
how does that go to clinical practice, I was kind of addressing that. But I totally understand the clinical trials, but it does then translate to clinical practice, which is decided by local and state as well as agency policies.
Yeah, I think Bathsheba's point is excellent, and it really addresses Ruth's question. Other questions?
So we have a question from Slido, which says, not all adolescents meet this category mentioned. Therefore, how do you determine the adolescents to be included in the research?
So for those two particular trials, then the ages were 15 to 17 and 18 to 22. They had to be high risk. And that was determined by an ACASI survey. So what they would do would be to answer questions in terms of screening questions that their response was anonymous. So the person taking the survey didn't know how they answered, so that they would be honest about their risk. And high risk, some of the criteria were having more than 3 partners in the last month, receptive anal intercourse, condomless receptive anal intercourse with someone with unknown or HIV positive, and sex for money, things like that. And so they were very high risk young people.
And then one more last one from Slido. Will another stem cell study be possible?
That's a good question. I think The answer is probably yes. I don't think stem cell transplant as part of the cure strategy is done yet. And there's certainly a number of groups still investigating this as a possible to restore the immune system.
I mean, the challenge with a transplant approach where you kill someone's immune system, you kill the good with the bad, right? The bad being the HIV infected cells, and then you wipe out the immune system, and then you repopulate it with donor or their own cells, and then they regrow an immune system basically from scratch. This is sort of what happened with the Berlin patient with a few twists in it, and it cured him. The problem is a bone marrow transplant by itself, if it's from an unrelated donor, has about a 10% mortality. Let alone the fact that it costs hundreds and hundreds of thousands of dollars. 1 in 10 people who get a bone marrow transplant will die from the bone marrow transplant. So it's not a simple, safe procedure. So if you talk about treating HIV or curing HIV with this process, I don't know, I think I'd rather take ARVs for a while longer before you put me through that, a 1 in 10 chance of dying. So there, you know, it's a great concept, but it's really, really still concept and very much in the realm of research.
My name is Robert Nunez. In terms of the research for the immunotherapy, what are, I guess, the next steps for it? You talk about your work being done in a lab.
Yeah, so for
what's
us,
the future for that?
the next step is to do some animal studies. I will say that the collaborator Catherine Bollard, who's helped us with the T cell expansion part, she already has approval for taking the unmodified HIV-specific T cells and introducing them back into patients. So that is, I believe, already started to look at safety questions.
So we have a question from Slido, which says, how can we make it easier for newly graduated clinical practitioners to break into the field of HIV care? What contacts are there?
We're recruiting for infectious disease physicians at Baylor College of Medicine right now.
And we need a research coordinator. People email a lot. So if this is a particular person, you can have my email.
My name is Kenneth Wirch. I am a social worker with UT Health. And my question is about viral suppression with the those that were perinatally infected who gave birth and they had issues with viral suppression after having their baby. Did you find, or were you able to determine if these mothers had an ongoing issue with viral suppression when they were adolescents themselves? And were there any kind of interventions to help address that?
Yeah, a lot of these patients, so we use that cutoff of 400 for the viral load because some of it is older data for their pregnancies. But a lot of them had adherence issues growing up. For the younger kids, there's the medications aren't very tolerable. You know, the liquid Kaletra, we still use it a lot, and that kind of thing. So there is resistance there. Lots of growing up with partial regimens for these older women. So they have had adherence problems. Depression was mentioned. Depression is not uncommon either. I think we haven't teased out all the different factors yet that is leading them to fail postpartum. But I think also because they did well during their pregnancy, they were able to do it. It also has to do with their social situation and the baby care as well. I don't know if I answered your specific question. Okay.
Regarding the study that was done at Rice, Dr. Chianta, I had a question that kind of concerned me a little bit. There was a comment made earlier that if that child who's on some parent's insurance policy is tested or receives a drug or so, that the parent will find out that the child was doing that. And that was a little bothersome to me because I heard that the study was a national study, correct? And so it seems to me that HIPAA, being a federal mandated policy, on the cover of our pamphlets it says from research to the real world. And so you were saying that there's a delineation that's made between research and clinical. However, does HIPAA not protect someone at any level involved in the healthcare system from having their information protected, their PHI, et cetera? And what steps from a healthcare body would need to be taken to shape policy that allows for children's parents not to find out if they were interested in taking these types of drugs? Because it seems to me in my actual profession of linking individuals to care, minors are concerned about their parents finding out, which could be a factor of deterring them from staying in care or to going through with a certain procedure. So is there a think tank already been developed to actually shape public policy to allow students or children who are going to be on their parents' insurance until age 26 for their parents not to find out what they're doing at the doctor's office?
So I just wanted to make the distinction between clinical care and study participation. So patients who are on the study, nothing is charged to their insurance for study procedures. That's paid for by the study. So that's separate, and then I'll let Dr. Moore talk about policy.
Yeah, so HIPAA does apply if you're a covered entity and you're engaging in research, right? You do, and that's why, part of your IRB application is how are you going to protect the confidentiality. So as my colleague mentioned, yes, research does translate to clinical practice, but the laws that regulate research are very different than the laws that regulate clinical practice. HIPAA is one that, because of confidentiality, it does, HIPAA, if you're a covered entity, you fall under HIPAA. So HIPAA is really a law that regulates the business of, the organization, right? So Baylor would be like a covered entity under HIPAA. And so any activity that Baylor engages in needs to align with HIPAA because Baylor is a covered entity under HIPAA. So whether it's research or clinical or whatever activity in the conduct of whatever business they do, HIPAA does apply. So the research, there's additional laws and regulations that regulate research and confidentiality and informed consent and that process. And then there's the HIPAA regulations and then ethical guidance around confidentiality and protection of patients. And so what was referenced earlier, and I'm sorry if this has created confusion, this blending or trying to hypothesize what happens, in other, clinical situations, but is that if in a clinical practice, not research, not related to research, a patient who's a minor shows up at their doctor's office and they want to get, the PrEP, then that doctor, yes, that patient, doctor, privilege, et cetera, hospital policy would protect that information. So the doctor wouldn't call mom and dad and say, hey, I saw Mary Ann in clinic today and she got PrEP. But what happens is that if you ever notice when you go to the doctor, there's this long legalese form that you sign that authorizes you to disclose all your private health information to the insurer to pay for that visit. So if that minor signs that, they now have waived their right to that protection. So that insurer who now receives the clinical information and knows about the visit, when any of you who have health insurance, that insurer then will send a disclosure, right, of all the things, the claims that it paid. So it's not an intentional, like, overt act of, you know, breaking the confidentiality, but basically, you know, in order to get compensated for the minor to pay for the visit, if they use mom and dad's insurance, then they had to sign away their rights to that protection. So the minor just has to pay out of pocket if they don't want mom and dad to know.
HIPAA says the information's confidential, but you can share the information if it's related to paying the bills, because the people who pay the bills need to know what they're paying for. And that opens the door to the insurance company getting the information, which they need to monitor, you know, payments and care and stuff. But it does create this back door, and so that's a tough one. It is a policy question, but no one did anything wrong according to the law here. It's just there's these unintended consequences. Okay, we're at the point of a break. We're going to break until 4:10, where there's another door prize, and then start with the last three sets of talks at 4:15. These are three outstanding talks. Another round of applause for the speakers. All right, guys, let's get going again. So we're at the home stretch. Three more presentations, all great. I think you're going to love them all. So the first is a topic that always people are always interested in, and whenever on the surveys each year about aging with HIV. So this is Dr. Rajagopal Shaker, who's associate professor at Baylor, and he's going to present some data. What's the formal title here? N-acetylcysteine and glycine supplementation in HIV patients rapidly improves glutathione levels, mitochondrial function, muscle strength, insulin resistance, inflammation, and lowers body fat. I've seen this data. I've seen it in a couple different forms. Really cool stuff. Small studies. This is not like ready for clinical prime time here, but very, very cool data. Shaker.
Thank you, Tom. Good afternoon, everybody. So you heard the long title, so without much ado, let me start with a little bit of a background. So I want to introduce you to mitochondria. These are tiny little engines inside every single cell in our body. So our body is made up of many different organs. We have a brain, we have a heart, we have a liver, we have lungs, we have kidneys. Each organ has a function to perform. If you take an organ, it's made of billions of tiny little cells. Each cell needs energy to do its job, and this energy comes from tiny little engines called mitochondria. So mitochondria produce energy, and they do that by burning fuel, and these fuels are primarily fat and sugar from the food that we eat, and they give us life-giving energy. Quite simply, mitochondria is equal to energy, which means it's life. All right? So problem is that mitochondria malfunction in patients infected with HIV, and they're not able to burn one source of fuel very well, which is fatty acids or fat, and we simply don't know why. Mitochondria also malfunction in other conditions, like, for example, type 2 diabetes in people who grow older, and again, medical science does not know how to fix mitochondrial function. Patients with HIV are also at risk at having, for example, muscle weakness, increased belly fat accumulation, insulin resistance, which is a kind of pre-diabetic condition, inflammation, which is harmful tissue irritation. And then the question is it possible that these defects could be linked to mitochondrial abnormalities? So mitochondria are burning all this fuel to make energy, right? And so think of the mitochondria almost like the engine of your motor vehicle. So we put in fuel, gasoline in our car, that goes to the engine, that's burnt in the engine, that creates energy that drives the car forward. Pretty much the same way the food we eat is broken down into its nutrients that goes to these mitochondria that burn it to give energy that drives the cell forward. Anytime you burn anything, you're left with toxic waste. If you burn wood in a fireplace, you get nice heat and warmth. but you're left behind with ash, right? And you have smoke that comes out the chimney. Similarly, in a motor vehicle, if you burn gasoline, you have smoke coming out the tailpipe, and all the dirty stuff is caught by the oil filter. If you've seen a clean and a dirty oil filter, you know what I'm talking about. In our bodies, we don't breathe out smoke. We make carbon dioxide that we breathe out. And a lot of these toxic material that's produced are called free radicals. They're also called oxidants. And anything that neutralizes an oxidant is an antioxidant. And turns out, you can go to your grocery stores, you can go to Kroger or Walmart, and you'll see antioxidants plastered everywhere. I turned on my TV the other day, and I found pomegranates and blueberries jumping around, right? The word is all over the place. But what you probably don't know is the most powerful antioxidants are made for free in our bodies every day, all right? And the biggest antioxidant the body sort of entrusted itself to is called glutathione. So it turns out mitochondria need a lot of glutathione to be healthy. But glutathione levels are very low in HIV patients, and we don't know why. So we started off, so why did we do the study? We wanted to answer a few questions and understand how this works and whether it can make a difference in people. So we did the study to find out why is glutathione levels deficient in HIV-infected patients. Is it possible to correct glutathione deficiency? Can mitochondria be improved when these glutathione levels get better? And therefore, is it possible to maybe increase muscle strength, lower belly fat and belly size, improve insulin resistance, lower inflammation? These are all important questions. So what did we do? We had a very small study. It's a pilot study. And in this, we had 8 HIV-infected men in whom we measured. These are otherwise healthy folks with suppressed viral loads, good CD4 counts, no opportunistic infections, no hospitalizations in the past few months, no diabetes, no liver disease, no kidney disease, as healthy as can be. And these are folks who were in the mid-50s. So we measured blood levels of glutathione, which were low. We measured their mitochondrial function. We measured muscle strength. We measured insulin resistance and inflammation. And as part of our studies, we found that clearly HIV patients had very low levels of glutathione. That's not surprising. Others have reported it, and that's what we found as well. But what we also found is that this glutathione is a protein that's made of 3 smaller building block proteins called amino acids. And 2 of these amino acids are called cysteine and glycine, and they were in very short supply. What is curious or interesting about cysteine and glycine is we normally don't get it from the food we eat. The body makes it on its own. It's considered so important that the body doesn't outsource the need for cysteine and glycine to dietary sources, but we make it on our own. And the body wasn't making enough, so there was a deficiency. So what we did next was we said, okay, if cysteine and glycine levels are low, is it possible to give it to patients in the form of a supplement? So we had this supplemented in capsules, and we provided this out to our patients for just 2 weeks, 14 days. And we gave the cysteine as N-acetylcysteine because cysteine by itself gets degraded in the gut. And we gave glycine as it is. So this is something we call NAT-GLY in my team. And we did this for 2 weeks, and we wanted to know how it made a difference in people. So what we found next is what I'm going to present to you is some data in the form of graphs, which shows you the effect of NAT-GLY supplementation in HIV-infected patients. So the first thing that you can see, this is the glutathione level of age-matched historical controls. So in my lab, I am interested. I've been working on glutathione and how it affects human health and disease for around 20 years. I work not only in HIV-infected patients, I also work in aging and geriatrics and issues therein, which are linked to mitochondrial function and glutathione. And so when I look at age-related complications that are affecting HIV, for example, accelerated aging, I've worked in HIV for about 20 years, so I'm able to have 2 different tracks come together. So one of the things I have through a lot of these research is that we have historical controls who are HIV negative. And so these are people who are the same age as these patients we studied, and you find that the glutathione levels is less than half. 2 weeks later, it starts coming up nicely, and it does not completely normalize, probably because 2 weeks is too short a period of time for it to come all the way to normal, and we need to do longer studies. But what happens to mitochondrial function? What we found is that in the fasted state, the fuel of choice in the body is fatty acids, right? When you have your dinner, you go to sleep, you're fasted overnight, and you wake up in the morning, the body is primarily burning fatty acids as a source of fuel because the gut is empty, the belly is empty. And that's exactly what you find in the HIV negatives. But you find in the HIV patients here, they're not burning enough, and that is the mitochondrial defect. But what is really, really cool is that 2 weeks later, as the glutathione normalizes, the mitochondria starts, the function starts coming up very well. Now, we published other papers where we've looked at animal studies in mice, where we find that if you take young mice and if you drop their glutathione, these are otherwise healthy young mice, and if you rapidly deplete their glutathione, their mitochondria is not able to burn fat very well. If you take naturally aging old mice, which have both a concomitance of glutathione deficiency and impaired mitochondrial ability to burn fat, and if you correct the glutathione, amazingly the mitochondria recover. And here you find the mitochondria recovers as well. And this is the muscle strength you would expect in a 55-year-old person without HIV, and this is where the HIV patient is. And in fact, this accelerated physical decline with muscle loss of muscle strength is one of the components of what is being called accelerated aging. And you find that 2 weeks later, that muscle strength is beginning to come up very, very nicely. If you look at body weight and belly size, the body weight comes down significantly. But what was really interesting is they lost about 3.5 pounds of total body fat in that 2-week period. And they lost 1 inch of the waist circumference. So that seems to suggest that a lot of the fat loss is coming out of the belly. I would caution you that this is a small pilot in 8 people. As Tom said, not ready for prime time yet, but clearly supports the need for more work. Every time I give this talk, people come later and say, where do I get my hands on this? So, all right. Insulin, if you look at it, there's a 31% improvement in what we call insulin sensitivity. That means the body's own insulin is working a whole lot better, taking them several steps away from becoming diabetic. If you look at inflammation, the harmful irritation, we measured something called C-reactive protein, and we found a 55% decline. C-reactive protein is an interesting biomarker because it's strongly linked to the risk of having heart disease. In fact, cardiologists, and if you read the literature, if you have a CRP value more than 3, that's highly undesirable. A highly desirable CRP is a value less than 1 point, okay? And you find that these patients are more than 3 points, and they come down by 55% in 2 weeks. The biggest study that studied this is something called the Jupiter trial, where they studied close to 2,000 people over 2 years of time, and they find that with a statin, it comes down by about 25%. We have just 8 people, but within 2 weeks, we have a 55% drop. And what I will tell you is that I'll show you the end. I don't have data. I can't share it yet because it's not published, but we just finished another study where we find identical results in HIV patients. So this is real. And what we also find is another inflammatory marker called TNF-alpha, again a 19% decline. TNF-alpha is a very interesting inflammatory biomarker in that in HIV treatment naive patients, when the antiretrovirals are initiated and viral loads are suppressed, TNF-alpha doesn't seem to come down. So it's really hard to make this improve. And we are finding here that there's a 19% decline, which is truly exciting. So when you put it all together, we find a very nice improvement in glutathione levels, in mitochondrial function, in muscle strength, in belly size, in insulin resistance, and in inflammation. So again, although these results are from a small pilot study, the results do matter because mitochondrial decline, muscle weakness, excess belly fat, insulin resistance, inflammation, these are all very serious complications which are affecting HIV patients today. And we do not know why these problems occur, and we don't know how to fix them. And therefore, this study gives us interesting leads because our results mean that we can now begin to understand perhaps why these problems may be happening. Could they be linked to glutathione deficiency? And maybe how to correct them. And N-acetylcysteine and glycine supplementation, therefore, could play a helpful role in improving the health of people living with HIV. And as I alluded to, we just completed a second pilot study where we supplemented people, completely different set of people, not for 2 weeks, but for 12 weeks. In the data that I just showed you, we used comparators from previous studies we'd done, In this study, we had age, gender, and BMI matched uninfected controls. And we gave them the supplementation for 12 weeks, and we stopped it for 8 weeks and studied them again. And what we find is that the results confirm and extend the findings. So the first thing that happens is glutathione levels completely normalize. The measures of body physical function, we measure something called gait speed, which is a really important parameter of physical function in geriatric folks, and it's very, very low in HIV patients. That completely 100% normalizes to HIV and uninfected age-matched controls. Muscle strength comes up in both arms, 6-minute walk test, they improve, and thigh muscle strength by something we call chair right test comes up. Insulin resistance improves, inflammation improves. Pretty much everything you saw happens in a more profound way. So we will be publishing this in the next few months. But clearly, collectively, these studies seem to indicate that something very exciting may be happening here, and obviously supports the need for additional studies. So early days, but I have a lot of hope and excitement. So at this point, clearly, studies cannot be done without help from a lot of people. So firstly, I want to thank my patients from my clinic who took part in the studies. So huge thanks to the patients who took part. Some of my team members who helped make the study happen. Our Baylor Clinical Research Center nursing staff, I still call it the Baylor GCRC, which doesn't exist anymore because the study was done a few years ago. The second study, if I were to present it, will have a different set of people who helped me with that. And of course, I want to thank our CFAR, which was helpful in funding this little pilot study when it happened. So at this point, I'll stop, say thank you, and take any questions if that's okay.
Yeah, we have time for a couple questions. Thanks, Shaker.
I'm sorry? If the money comes.
Hi. So quick question. So why do you think it's glutathione correction that's bringing up all this sort of positive effect and not an overall cysteine correction, right? Levels in cysteine, because cysteine does a lot more.
Because cysteine, N-acetylcysteine has been around for a while, and none of the studies which have used NAC have shown any of this stuff. And in our animal studies, what we did is, this is unpublished data, we had we had 3 groups of young mice. In one group of mice, we gave them, these were control mice. Second group of mice, we gave them an inhibitor of glutathione synthesis. So this inhibitor prevents the body from making glutathione. In the third group, we gave the inhibitor of glutathione synthesis, and we gave the NAC and glycine. And we found that NAC and glycine by themselves have no effect. They seem to act only through glutathione. And so this clearly seems to be the mechanism. We have more than one animal study which clearly shows that.
That is interesting.
Is there a way for you to, or the field to be proactive with this and possibly begin treating, I'm not going to say kids, but maybe young adults with the, what is it, NAC-GLY? Mm-hmm. Would it be in their best interest, or would that possibly cause other problems down the line as they get older by introducing more of this into the body when they're younger? So in terms of introducing problems, I will tell you that, as I said, I have 2 parallel tracks. I work with old aging by itself, and I've been working with HIV by itself. The longest trial we've done in aging is a 24-week supplementation, and we've had no side effects. And this is a 12-week study. We have a randomized clinical trial, NIH-funded double-blind clinical trial in geriatric older people that's been going on 5 years. That'll finish this December, and we should have data next year. So that's a 16-week trial, and again, there's no side effects there. So far, I've not seen anything that's necessarily worrisome. On these folks who did it for 12 weeks, we have safety labs done every month where we measure the liver test, the kidney test, etc. And we had absolutely no problem. If anything, parameters improved. Not a single person dropped out. In fact, they came back asking for more. So whether it can
on
have...
that note, we need to move on.
Okay.
There'll be time to talk to Dr. Shaker, ask additional questions after the last 2 presentations. So thank you.
Thanks.
Excellent. So our next presenter is Dr. Elizabeth Chow. Dr. Chow is now full professor at Baylor College of Medicine and also is sees patients at Thomas Street. So she's going to talk about, what is she going to talk about? Sorry. Liz is going to talk about Kaposi sarcoma remaining high, unchanged in African American males in the southern U.S. Cancer statistic data. So Liz.
Hi, thanks for inviting me to present these data. We presented these data actually at the International AIDS meeting about a couple months ago in Amsterdam. I think it's really relevant, and it's sort of the opposite of Shaker's talk. He's bringing really good news, and this is sort of maybe not bad news, but more sobering news. And this is, again, opposite in that this is large total population data in the United States. I'm just going to say a couple words about HHV-8-associated Kaposi sarcoma. It's characterized by development of vascular nodules in the skin, and these are really common manifestations. You can see on this nodular lesions on the skin, in the mucosal tracts. They're often in the lung. And then here's another example of skin lesions. They are it's endemic to several areas of the world, including Africa and the Middle East and then around the Mediterranean. But mostly for the U.S., it's been very important in HIV disease and has really decreased since the arrival of antiretroviral therapy. So a lot of physicians don't see it anymore. So why we did the study was, as I said before, there has been a 1,000-fold decrease in Kaposi sarcoma in people living with HIV compared to prior to antiretroviral therapy. There's a 1,000-fold increase in patients living with HIV compared to the general population. But it's decreased by almost 85% here since 95, when antiretroviral therapy was widely introduced in the U.S. And the other reason is because we here in Houston, we're still seeing a lot of Kaposi sarcoma, and I was talking to my colleagues around the country, and they were not seeing very much, and so we wanted to see if what we were seeing here was really something that was different than the rest of the U.S. So we originally used the National Cancer Institute surveillance of epidemiology and end results, and at that time we did find that KS was higher. But that's only in these certain states. So that was California, Connecticut, Georgia, Hawaii, Louisiana, New Jersey, New Mexico, and Utah. What we did here was we furthered that line of research by specifically looking at age and race as well as geographic differences. And we used data from the full U.S., not just from SEER. So this is from the CDC's wide-ranging online data for epidemiologic research platform, or WONDER. And we looked at all incidence KS cases because KS is relatively rare in the U.S. Pretty much all cases between the ages of 20 to 54 will be related to HIV. And again, that's not 100% the case, but we've done previous studies that have validated it to be over 98% of cases in that age group are HIV related. So what we found was, first by age, we found that although in the 30 to 44 age range, there's been a really strong decrease in KS, there's really been not a lot of change in the 45 to 54 age range. And again, if I'll point you out to, these are annual percent changes, and then if they're bolded and there's an asterisk, it means that it's statistically significant to the p less than 0.05 value. So there's been a statistically significant increase in young men between ages 20 to 29. And statistically significant decrease in the 30 to 44, but no change in the 45 to 54 age group. Then when we looked at region, we do see that actually in all regions there are statistically significant decreases of Kaposi sarcoma. However, you can see that the South has had a lower slope in that decrease, and now the incidence rates are the highest among all the regions. When we look specifically at a metropolitan area, we found that these are the top 5 metropolitan areas in the year 2000. And we look to see what the trends have been, and you can see that Atlanta, Georgia has a non-statistically significant increase. So although the trend is upward, it's not statistically significant, but you can see that it's different than all the other metropolitan areas that have statistically significant decreases. And then we looked at African American men by region of the country. The join points are what the computer, in our modeling, we basically were able to see that the annual percent change actually was different over specific years. So I wouldn't pay much attention to the join points at this point, but to say that in the South, although there's a trend towards decreasing, it's not statistically significant. And really among all the regions, again, this is only African American men, among all the regions, the South really stands out because there has not been a statistically significant decrease. So I would say that in our southern neighbors, we are seeing just as much Kaposi sarcoma in African American men as we did in 2000. So here's a summary of our results and why this matters. So KS significantly increased in men aged 20 to 29 and really was unchanged among older men aged 45 to 54. Those are sort of stunning statistics given the previous finding of between 96 and 2000, an 85% decrease in the number of cases in the U.S. In African American men, there was no significant change overall in the U.S. I didn't present the data, but there's also a very statistical significant increase in this young age group among African American men. And then in the top 5 MSA KS incidents did increase, but not statistically significantly in Atlanta, Georgia. And now the incidence is twice that of New York City. And I'd just like to acknowledge my co-authors and project staff, and we received funding from CFAR, NCI, and then this was done with help from funds through the VA. Thanks.
Questions for Dr. Chow? So one thing that just to be crystal clear, KS is a cancer, just in case you missed that point. So it's not something it's not a minor disease. This is a major health threat to people who get it.
And it's associated with immunosufficiency. So one of the things that I would say is that the fact that Kaposi sarcoma, the trends of Kaposi sarcoma will follow advanced, sort of more advanced and late HIV diagnoses.
Thanks, Liz.
Sure.
So that my question actually follows that. Have you overlaid the rates of new diagnoses in different age population to see if the rates that we see in the South for KS actually is following the increasing rates of HIV new diagnoses among those populations?
Yeah, we actually did have a map, but it was a little bit, it was kind of a lot of data. So in our, when we publish the paper, we will have a map that does show that HIV incidence rates are sort of continuing to decrease pretty much in all regions, and KS is still spotty. So it implies that there's a specific KSHV viral component to it. But also what we overlay was new diagnosis of HIV and not necessarily advanced AIDS, so that's also a possibility. Yes.
You were saying that when you started this, it was because you hadn't seen so much of it. So your data is national, but do you have any local data?
Yeah, so Tom actually said. So we did look at Houston, Texas. It actually didn't make it into that's so why it wasn't in one of the MSAs. It didn't make it into the top 5 in 2000. When we redid the analyses to look at the top 5 MSAs in 2014, the top 3 are Atlanta, Georgia, Dallas, Texas, Houston, Texas. So we're among the top.
So
yeah.
this is top in terms of basically number of cases of KS per population.
Incidence rates, right, per population.
Yeah. So Houston could be in this if she chose to structure the study a little differently. Houston would be in there, but the way she set it up with where they were back in 2000, was it 2000, Liz?
Yeah, it was 2000.
Houston was not at the top, but it just, again, reflects the growing rate of this, or at least the not declining rate of this in the South.
Right. So you saw all those other MSAs that were decreasing really quickly. New York, LA, those rates, San Francisco, the rates of KS are decreasing. Over this last 14 years have decreased, and so now, and Atlanta was like number 3 or number 4. So Atlanta has moved up to number 1, and then actually, so now there are different MSAs that are the top 5. And I think all of them, I forgot number 4 and number 5, but I think all of them are in the South now. Yes.
I'm sorry. Can you hear me? Okay.
Yes.
Question. Do you think diet or environmental factors play a role in any of this data that you just presented? And next question, are you talking about all the southern states?
We used a definition of the South by the census. I don't have, unfortunately, I should have put a map up, but basically it's sort of below the mid-Atlantic. It's anything that's south that's below the mid-Atlantic, but Texas is included.
South Carolina, the whole
yes.
South Carolina,
right,
along that route.
Right, and the western edge is Texas.
But what does diet, or like I said, does diet and environmental factors? Have you thought about that? Is that possible? I guess that's what I'm trying to figure out.
Studies have, a lot of studies have been around Kaposi sarcoma, especially earlier in the epidemic, before antiretroviral therapy, because it was so common, and diet and environmental factors did not come into play at that time. But again, the influence of immunosuppression is so important in this disease that it could mask a lot of other things. And so making sure that the immune system and diet plays into that, right? So you have to eat well to help your immune system be as healthy as possible. So that's possible, but just, it's what we have to measure our immune system is primarily CD4 count. And so that's really the most important predictor of whether or not you get this disease.
Yeah, you rarely see this in someone whose CD4 count is high.
The only reason I mentioned diet, because if you're in the South, people eat a little bit differently in other parts of the country. Not to say that we all eat bad, but I'm just speculating. I can see it could be some issues, but like you say, you didn't see a correlation, but
you
just
might
curious.
Want to explicitly mention the HHV8. That might be news.
People, you're going to eat a lot of fried food here in the South, and it could affect a lot of different things, I think. But,
sure. But I think so just going back to the immunosuppression piece,
yes.
so as Tom said, it's this virus is mediated by this cancer is mediated primarily by a virus. It's caused by a virus called human herpes virus 8, or Kaposi sarcoma herpes virus.
Right.
And so, as I said earlier on, it's endemic to certain parts of the world, like Africa and around the Mediterranean. And so it could be related also to the fact that in the South there may be higher rate. They haven't shown that in a lot of the epidemiologic studies. So the prevalence of human herpes virus 8 infection in the general population in the U.S. is usually less than 10%. So whereas in Africa, by the age of 10, 60 to 70% of the children have already been infected with HHV8, just to give you a sense that this can be very regionally involved. The other thing is that I want to just bring back to the point of the importance of immunosuppression. So this disease can also be seen, is also primarily seen in people who are immunosuppressed for other reasons that are not HIV. So transplant-related Kaposi sarcoma is actually really common. So again, if you have a lot of transplant, and we didn't know if these people had transplants or not. So if you had a lot of transplants, it might have fallen into this data. But again, immunosuppression is so strongly associated, and then there's the virus. It's hard to study these other factors because they're just so strongly related to those other two.
Thank you, Dr. Chow. We're going to move on to Dr. Flash, and then we'll have the panel. So our last presentation is Dr. Charlene Flash. Dr. Flash is the Associate Chief Medical Officer at Legacy. Oh, it's late in the day. Legacy Community Health Services and an adjunct faculty member at Baylor College of Medicine. And she is going to talk about HIV risk perception, pre-exposure prophylaxis perspectives, and its use among women, issues of disclosure and trust. Just hit next, and your slides will come up. There you go.
All right, great. Good evening, everyone. All right. I know I'm the last one, but you guys have to wake up. Okay, because I need some energy to get me through the day. So I'm going to talk with you a little bit about HIV risk perception and PrEP perspectives among U.S. women and Black women in particular. So the reason we did this study is when you look at the general population of the United States, I'll draw your attention to the pie on the left-hand side, you'll see that 12% of the U.S. population is comprised of people who are Black or African American. I'll draw your attention to the center pie. When you think about the proportion of the HIV epidemic, which is comprised of people of color and of Black and African American people in particular, you'll see there's a disproportionate number of Black and African American people who have HIV infection in the United States. So here it's 44% when you look at all people. If I were to put up a slide that just had women on it, Black women amongst all women in the United States make up almost 66% of infections. Now, if we, as we think about PrEP, our new and exciting way of preventing HIV and its dissemination in the population of the United States, you would say, well, PrEP should go at least with a population distribution. So at least we'd say there should be 12% of prescriptions happening in this population. Others would say that because this population is disproportionately infected, maybe most PrEP prescriptions should go into this population. Unfortunately, that's not what's happening. When you look at the third pie, all the way to the far end by the door, you'll see that only 10% of prescriptions that were written for PrEP at the time of this data were written amongst Black and African American people. And if I were to put up a slide that just showed women, the numbers would be much, much smaller. So recognizing that women of color bear disproportionate risk of HIV infection in the United States, yet are not receiving PrEP prescriptions at the same rate, that is part of what caused us to want to do this study and continue this investigation. So what we did was we initiated a prospective cohort study of patients who presented to Thomas Street Health Center and Harris Health System. We recruited 271 individuals who presented for walk-up HIV point-of-care testing, and we screened them for HIV risk. 183 of those patients followed up for MD evaluation, saying that, yes, I agree that I'm at risk and I would like to investigate more about PrEP and find out about that. And they also received HIV education and adherence counseling, and a subset of those received a prescription. Now, of that sample of 271 individuals, we restricted the sample to just the women that we enrolled, and that was 109 women. We collected mixed methods data, so that means we collected different types of data, including both survey data as well as doing key informant interviews. So we actually talked to these women about their perspectives on HIV, on their own HIV risk, on different modalities of HIV prevention, and as well as what they thought the impact of PrEP might be upon their sexual relationships. And we followed these women over time, so we did baseline and interim assessments of these individuals. Now, what we found was that of the 109 women that we surveyed and did these key informant interviews on, nearly 70% of the women we recruited were Black or African American, about 20% were Hispanic, and about 10% were white, non-Hispanic, and 2% Native American. Half the women were working either full or part-time, the other half were unemployed or students. Nearly 80% of the population we recruited were making less than $25,000 per year. And although the majority, 70%, were stably housed, most participants had no health insurance. So nearly 56% or nearly 60% did not have health insurance. Now recall that they were recruited from Harris Health System, so even though they did not have health insurance, they, through the Harris Health Eligibility System, would be able to access health care through the Harris Health Eligibility Program. Now when we ask these individual women information about their personal lives, in the past 6 months, 80% of them had had a male sexual partner, 2% or nearly 3% had had a female partner, and 1 had had a trans partner. And there was a fair amount of these women, about 15%, who had had no sexual partner in the prior 6 months. Now, when you look at what their partnership status was, now this is a different question than who you're having sex with, because you might have someone who is your partner, but it's not someone that you're actually having intercourse with, and I'll talk a little bit more about that as we move forward. But about 27% said, I have a steady partner, and 32% said, I have a spouse. Only 26% said, I have multiple partners over the prior 6 months, and 20% described their partners as casual partners. Now, of these women who you'll see was a relatively at-risk, socioeconomically disadvantaged population, a relatively small subset actually said, I would like to attend an HIV prevention program appointment. Only 27 of the women did. And a smaller subset of those women actually started PrEP, so an N of 20. So let's look a bit more at that. So here we see some of that data about people's partnership status. I'll draw you down to the third line on this chart where it talks about whether or not these individuals were using condoms when they have sexual intercourse. And what we found is that about 20% of women said, yes, I always use a condom when I'm having intercourse. And 32% said, yes, I do sometimes or most of the time or rarely, but not all the time. Sometimes I use condoms. But 46% of women said, no, I never use condoms during sexual intercourse. Now, the other part that's interesting about this is because these women were recruited from the walk-up testing program that's housed in one of the largest freestanding HIV clinics in the country, it's not surprising that a large proportion of these women actually had sexual partners that were known to be HIV positive. So 39% of these women said that, yes, they had at least one of their sexual partners that was indeed HIV positive. So of these women, we also asked, they received a very brief information session about defining what PrEP is, and then they were asked, would you be interested in PrEP? And 86% of the 109 women said yes, they would be interested in PrEP. When asked about concerns, 37% had one concern, 40% had multiple concerns, and only 20% said, no, I don't have concerns. All right, so you have a population that's socioeconomically disadvantaged, who has risk, whether it's number of sexual partners or the fact that they have a known HIV positive partner. Most of them are saying that they are PrEP interested, and yet only 27 of them actually followed up for an appointment. So in talking to these women and getting their stories, it helps elucidate this challenge a little bit more. So first, one thing that was quite interesting was this issue of disclosure. So these are in the women's own words. The first was a 25-year-old married woman with an HIV-positive spouse, and she said, well, of course, nobody's going to say, oh, hi, my name is such and such, and I have HIV. But eventually, he did tell me. He didn't want to tell me, but he did. Another 29-year-old woman who was in a marriage where her partner was HIV positive said, but recently he went to jail. Next thing I know, I get a phone call, and he's saying that he's not really telling me, he's beating around the bush. I had to pull it out of him for him to tell me that he was positive, meaning HIV positive. So then in terms of risk perception, a 41-year-old married woman said, well, I heard that my ex-husband was with someone that did have HIV. And it was like, let me get me checked out. So for those who actually did avail themselves of PrEP, we did find that PrEP did impact their relationships. So one 25-year-old woman said, yes, because when we did have sex and I wasn't on the medicine, but we used condoms, I wasn't able to, like, you know, how you're able to like get into it and be relaxed with no worries. I was like, okay, hurry up, come on, and then let me check the condom and make sure it didn't break. Now it's a little more enjoyable. It's not like me worrying. A 30-year-old single woman said, well, he wanted me to be on PrEP because he wants to protect me as well, because we have children. So the most important thing is keeping him healthy and keeping me healthy so we can be better parents. So in thinking about what our results mean and why this matters, we recognize that regardless of what our risk perception of these patients, what we perceived their risk to be was, it was important for these individual women to self-assess risk if they were indeed going to get on PrEP. And that for women, this posed a challenge because oftentimes their risk was dictated by what their partner, whether or not their partner was HIV positive, and then this issue of, is he going to tell me yes or no, and what's the driving force for him to actually disclose the status? So women described these barriers to disclosure, and they described this perception of personal risk that was wrapped up in whether or not their partner had disclosed or whether or not the person down the street disclosed on behalf of their partner. But this was part of what was needed for them to uptake PrEP, for them to actually see their risk as being real, whether or not they had other risk factors that we as scientists or clinicians would think would be risk factors for HIV. Overall, in this population, there was very limited condom use. Most women felt positively about the idea of PrEP, but PrEP interest did not always translate to program enrollment with adequate linkage to care or even to PrEP use for those who actually showed up for the program. Nonetheless, women who actually started PrEP, they described positive experiences, both with the medication in their sexual relationship and actually with the process of being involved in the program. So when I think about the import of this, it's a little snapshot into what heavy lift has to be done in order for us to fix that very first slide, right? If these black women are comprising 66% of the population of HIV-infected women in the United States, we have a lot of work to do in terms of raising their understanding of their HIV risk, their risk perception, getting them to have their partner disclose their status, and then having them move forward to access an available and effective mode of prevention. So I'd like to acknowledge my research mentors and my funders, Gilead Sciences and the NIH. I'd like to acknowledge Harris Health System and our study participants, as well as my many collaborators and research coordinators who participated and helped with this research. I'd like to entertain your questions.
So I guess just a couple parts to just throw out. So what were some of the concerns about getting onto PrEP you mentioned by these women? And also, what were some of the, I guess, the barriers to women accessing PrEP? I think you said there was 26% for follow-up and getting onto PrEP. Or what do you think from this research was that barrier for a higher follow-up?
Absolutely. So your question is, what are the barriers to getting onto PrEP, and what were some of the things, the misgivings that the women had about getting on PrEP? So the barriers are multiple. And so for some of these women, it was just going to an appointment, whether it's my work, transportation, kids, who's watching the baby, does my partner let me go, all of those types of actually getting to the appointment barriers. For many of the women, it was that even though PrEP sounded like a good idea when you were having that individual conversation, that the leap from it sounds like a good idea, but I think I individually am at high enough risk where I want to take a pill once a day, that was a big leap, except for women who said, I know my partner's HIV positive. women had a variety of concerns. Some had one concern and some had many. The most common concerns were about side effects. Is it going to make my hair fall out, make my kidney fail, make my liver fail? I don't like taking pills. So multiple pill-related factors. There was a smaller proportion than we expected that had issues with medical mistrust. Are you giving me HIV with this pill? That did come up time and again, but it was not as common as you would think, as I thought that it would be, that you're trying to poison me with this pill, or you're trying to give me HIV with this pill, or those issues of medical mistrust. Perhaps that is the relationship that they developed with the navigator who was doing their HIV test, that there was a trust relationship there. That wasn't as much, but there were some pill-related barriers. But I'd say primarily it was the women self-attributing risk and being able to figure out whether or not their partner was positive, and without that knowledge, it was often not a big enough leap for them to say, I'm going to go ahead and do the follow-up to take this one pill once a day. Great question. Thank you.
Good evening. Thank you, Dr. Flash. That was good. But I have a concern. I read recently how Black or African American women, the doctors, in fact, it was on TV, that they don't listen to us as well, or don't take our hurt as serious as they do the others. And I wanted to know, how did that relate to your research, or have you experienced women of color telling you they just don't listen to us?
That is an excellent question. It was not part of this project, but the next. And looking at that, I will keep it brief though, so you can pull the hook. So, in thinking about PrEP dissemination amongst women and amongst people of color, the issues of stigma, racism, and poor communication between patient and provider are commonplace. And so there is what I have found to be this disconnect between what the patient's expectation is and what the provider expectation is. And so providers might say, hey, I want someone to walk into my office and say, I think I'm at risk and I'd like PrEP. Please prescribe it to me. Whereas the patient feels as though the provider should assess me and then tell me that I should take PrEP if that's what's appropriate for me. So those two things are worlds apart. Each party is waiting for the other person to tell them that this is a good thing to do. Now, whether or not this happens more, and whether it's really that it's the provider doesn't see this patient or hear what their needs are, or whether it's that certain populations are more self-empowered to come in and say, this is what I want, right? That part I haven't figured out yet, because I think there is a piece of that where certain patients will come in and say, I read about this cool thing and I believe that I'm at risk. Can you please give it to me? And other patients don't have that kind of relationship with their providers where they don't come in and ask for things. They offer their symptoms and expect the professional to give them advice. So it's complicated. You're welcome.
Can we hold your thought? We're going to have time for more questions, but I want to bring Dr. Chow and Dr. Shaker back up so that people can address questions to all of them. But your question for Dr. Flash.
So my question is actually going to piggyback off of the one that you just answered, simply because in my line of work, I typically deal with, my focus population is young Black gay men. However, just because I'm aware of PrEP, I'm a user of PrEP, I typically try to get people in my social circles to be able to, be in the know. And so oftentimes I have issues having conversations with women about the use of PrEP and how the benefits that they can have, simply because if I go and give them this information, right, and then I tell them to then go and talk to their providers to see if they can be able to get any more information from them, there is a lack of follow-through when it comes to the provider standpoint, right? And so my question is, what is it that you think, just based on your experience with working with women, what is it that we could do to be able to give them a little bit of a better foundation, so if they do not know how to advocate for themselves, then they can go in a little bit more prepared to be able to ask questions that they maybe not would have asked prior to. Okay. Does that make sense?
It does. So
okay.
It sounds like what you're saying is that in your work in the community, as you identify women who could avail themselves of PrEP, your concern is that when they go into the provider, they still might not get it.
Sure.
Okay. And so I think that the solution to this is two-pronged. That one, it's keep doing the good work of informing women that this is available, because many women just had never heard of it. So the information is incredibly important. And then encourage them to ask their providers. Patients asking providers for things is actually a much stronger motivator than a patient might think, right? And so if the patient goes in and says, hey, I think I'm at risk and I want this or I need this, even the provider who is a late adopter often will go and say, well, I don't know anything about that, but let me go research it and figure it out. Or let me refer you to someone that can help you, because the clinician perspective is that I want to help this person, even if this is something that I've never heard of, okay? The other portion, though, is that we have to do some focused work with providers to educate them about PrEP. Even though it was FDA approved in 2012, not all people who can prescribe PrEP know about PrEP or have the frame that this is not just something that's for gay men, but that it can also be something to help women who are at risk as well. And so we have to do that focused work in addition.
Do you want to talk about the PrEP locator? Just mention that.
Yes, and so there is something called preplocator.org, which is a website where you can put in your zip code and it will identify spaces where you can find providers who are willing to prescribe PrEP. Now, for your women issue, sometimes these prescribers willing to prescribe PrEP are most used to prescribing to men, and so you will find a fair number of, men-focused clinics. But if there's a woman who needs it, she should go where people are available to them. I've had men go to Planned Parenthood to get PrEP in certain communities where that's the only place they could get it. So it goes both ways. You're welcome.
I have a question for both Dr. Flash and Dr. Sakar. Dr. Flash, so I understand that women are hesitant to ask their partner about HIV status, or their partners are hesitant to disclose their status to them. What did these women feel about HIV testing before having sex? Like, what were their views on getting tested with partners, and how resistant were they to that? Or how inconvenient was it?
So I did not ask that question of this group of women. I did ask that question of a group of women that I did focus groups on in Boston. And those women said, hey, I would love to get tested with my partner. In fact, one woman imagined a scale that she would have next to the bed. And so before they got in bed, they would both stand on the scale and it would tell you if you're HIV positive or not. That woman was prescient and could have made lots of money, because now you can go into CVS or Walmart or Walgreens or wherever you want to go, and you can get a home HIV test kit. So that imagining that she had, 7 years ago is almost a reality. We just don't have the scale part. So testing is an important element of this as well. But I still tell people, whether providers or patients, it's still important to talk to people, right? If you're going to do the most intimate thing on earth, you should be able to maybe talk about it a little bit beforehand. So I try to give that harm reduction education for women as well.
Thank
you.
And, question. And Dr. Sakar, why wasn't glutamate included in the regimen with the?
That's a great question, simply because the levels were not low. So most of the amino acids in the body, when they get broken down and they are degraded, they trace the pathway through glutamate. So we found no deficiency, so there was no need to replace it.
Hi, this question was for Dr. Flash. Dr. Flash, my question is related to social support. I'm right here. Yes, so in the population that elected to take the PrEP, did you see an association between increased social support and the increased likelihood that they would take the PrEP? And then I have a secondary question, or maybe a linked question. And then if not, do you think that active listening and empathy on the part of a healthcare professional could help to bolster that where social support was lacking?
So the question was whether or not women who had social supports were more likely to access PrEP, and do I think that a provider could be that support giver in the absence of support for the women. So although most of the women who availed themselves of PrEP were in some kind of relationship and with an HIV positive partner, sometimes that relationship was not one in which there was support. And in fact, it was more common than not that women felt that the staff, the multidisciplinary staff that served them in the program, that became their support system. And so some women just liked coming to the program because they said, God, this is a space where I can have a non-stigmatized discussion about the realities and complexities of having an HIV positive partner and the stigma that's related to that and how hard this is, not just about the PrEP part of it for them. And so I don't know that having support brought them there, but the support they received there kept them there. And even though the provider can be a compassionate figure, and I hope that patients found me to be a compassionate figure when I engaged with them, that we shouldn't underestimate the support of non-clinician support. And so for many of these women, it was what they received from the patient navigator or from the medical assistant or from the ancillary members of the clinical team that they felt was their support system and had an alliance with them that was stronger at times than what they had with the clinician.
So I have two questions from Slido. The first one is, why is KS more prevalent in African American men?
That's a really good question and complicated. And I think so. The data that we showed suggests that that the incidence in African American men is higher, but also the incidence of HIV is higher. And so these two epidemics are so linked in the U.S. So it's hard to know if there's anything outside of HIV that kind of that influences who gets Kaposi sarcoma.
And the second question from Slido is, does PrEP have a timeline as to how long one can use this medication?
Okay. So there's not a timeline, there's not a limitation for how long someone can use the medication. How long someone should use the medication is as long as they need it, so as long as their season of risk is. Unlike HIV, where you tell people that their treatment is for life, PrEP is for the period when you are at risk. And so if you are no longer in a space where you're vulnerable to HIV, then PrEP doesn't need to be a part of the equation for you.
Hi, I noticed, I remember that you said something about that the KS was regional. Was there certain parts that I didn't get to see the slide where it showed the illustration? I think I missed that part. But was there certain parts in Africa where it's more prevalent than other parts?
So in Africa, it's interesting. I mean, so sub-Saharan Africa has very, very high rates of Kaposi sarcoma. And it was also, there's more endemic Kaposi sarcoma also in Africa, again, related to the high prevalence of Kaposi sarcoma herpes virus in the population. So, but the countries that have the highest rates of Kaposi sarcoma are those that have the highest prevalence of HIV as well. So I can't, I'm sorry, I don't have the numbers of each of the different countries, but as a whole, sub-Saharan Africa has a much higher rate of Kaposi sarcoma than the U.S.
Hi, how are you? This question is actually for Dr. Flash. And while conducting your questionnaire, did you ask why the condom was not used during sexual intercourse? I wanted to know whether they just didn't want to use it or maybe they didn't have one available. I'm a community health worker, so I kind of want to know what the barriers were.
That's a great question. The question was, did we ask the women why condoms were not used? And so the way we investigated whether or not, or why not condoms were used was actually on the survey. I can tell you the kinds of questions we asked, but I can't tell you the answer for women because we haven't looked at the data for just that subpopulation. So the kinds of questions we asked were about whether or not one thought that condoms were uncomfortable or would impact the spontaneity of intercourse, whether or not you felt as though you could have the power in the relationship to navigate condom use, whether or not you felt as though your partner would think that you were unfaithful because you opted to utilize condom use. Across the board, patients said things like, I think my partner will think I'm unfaithful if I navigate condom use, or I just don't like the way it makes sex feel, were the most common things. We didn't have dialogue about condom use, which would probably be more robust, like just to actually have the conversation. But that's a great question. More data to come. I have to look at it more carefully. You're welcome.
To add about the last statement about the condom use, I think maybe some education on how to properly use condoms. I mean, maybe they don't know there's different options out there. There's things they can add to make them experience more pleasurable, I guess. But like maybe some education about preventative.
No, absolutely. So the comment was about more education about condoms, and so I don't want to leave the impression that when we prescribe PrEP, we just give someone a pill and leave it alone. It's packaged in a full prevention package. And so that package involves 4 things generally: helping people have dialogue about their partner status, whether they're HIV positive, negative, is the partner linked to care, things like that, navigating condom use. So use of lubricant, use of different, is it flavored, rib, whatever, different things about condoms, getting STD testing and treatment, and the importance of condoms with that to protect yourselves from STDs because that facilitates HIV transmission. And then the kind of sex one is having, whether it's anal, oral, and vaginal, and what position you're in for each of those in terms of how little or much risk you're at, depending on what type of intercourse you're having. And so harm reduction PrEP is part of the harm reduction strategy and is not, in my opinion, a standalone.
Hi, my question is for Dr. Chow. We have that 17-year-old newly diagnosed with AIDS. His CD4 count was 1 and his viral load was 2 million. And he has the KS on all extremities and on his buttocks too, and now it's spreading to his face. So I was wondering, how exactly is KS diagnosed? Because the doctor did a CNS culture, and he came back some type of staphylococcus something. And another pediatrician he saw told him it was flea bite infection.
I would send them to a dermatologist, and they need to biopsy the skin. It's a skin biopsy. It's a cancer, so a pathologist would make the diagnosis. In some places, you can kind of, people will, see it and kind of say, well, it looks like KS. Nobody will give him chemotherapy without a biopsy. Sometimes starting antiretroviral therapy antiretroviral therapy is the first line of therapy. So that person needs to be started on antiretroviral therapy. But sometimes it can make it worse because there's descriptions of iris or the inflammatory reaction after starting antiretroviral therapy, and it happens with opportunistic infections, and that includes Kaposi sarcoma. So if he gets worse when he starts his antiretroviral therapy, he needs to be immediately seen by an oncologist. But I would try to get him to a dermatologist or send him to see me.
Okay, because he's just started. He started the ART today. Today is his first day.
Okay, so yeah, watch. I would say to the physician that he needs to be watched kind of carefully, especially when it's already that kind of aggressive. Sometimes, it can. We've had the last couple months, we've had somebody admitted to Bentov because the lesion obstructed his airway and he needed an emergent trach. So and it's oftentimes when it's diffuse on the skin, it's often also in the viscera as well.
Okay, thank you.
And I would add that if you're not a doctor who sees people with HIV or thinks about people with HIV, you're not going to think about Kaposi sarcoma in a person who doesn't have who you don't know has HIV, because it's that strongly associated with HIV. So if they were seeing some general doctor who may be a great doctor but doesn't think about HIV or see people with HIV, they may not even think about KS as a potential diagnosis. So it's really important to get.
And even dermatologists
yeah.
don't. So, but that's the good thing about dermatologists is that they biopsy everything. Right. So.
All right. Well, that wraps up the question and answer
Maybe
portion. All right. One quick one.
Yeah, you brought up a question, Dr. Tom. You said if you don't know doctors that do HIV. My question to you was, is there a list, or you all listed that you serve the HIV community? And if so, where do we access that list?
So there is the PrEP locator also has HIV treatment locator function. So there are online resources to find that information. The Ryan White Planning Council produces a blue book, which is eventually supposed to be online. I don't know that it is yet. Is it, Stephen? Yeah? Okay. But if you look for the Ryan White Planning Council website, they can give you phone numbers to find out various HIV providers, especially those that don't require insurance, that can care for people who don't have any insurance. And what other resources are there, guys? That's right, yeah.
I was going to mention, the online version for the Blue Book, I gave you the website for the Planning Council is www.rwpchouston.org. And there's a left-hand panel that you'll see it says Blue Book. And click on that, it should give you that online version.
Yeah.
The app that will take you there.
So the Blue Book is the comprehensive. It's like the telephone book for anything HIV, all HIV services available in the county that are associated in some way, shape, or form with the Ryan White program. So it's a very comprehensive list. All right. Thank you to the last three presenters. Fantastic presentations. So Larry Roberts is going to make a few announcements, and then we're going to wrap this up almost exactly on time.
Hello all. Oh, okay. Before we close up, I just want to give a few thank yous and some minor housekeeping. And I have a list, so bear with me. Thank you to all the speakers for their contributions to the community, the research, and for sharing their work with us today. Okay. Thank you to the sponsors of the event, the Houston Health Department and Baylor College of Medicine and Dr. Tom Giordano. Thank you to all of the Houston Health Department volunteers. Thank you to our co-facilitators, Dr. Tom Giordano and Jeffrey Campbell. Thank you to my fellow planning committee for the event, Raven Bradley, Megan Cannon, Chelsea Frand, Camden Hallmark, Robert Ross, and Ricardo Mora. Our logistics coordinator, Mr. Barry Barnes. Thank you to the medical providers and organizations that assisted in getting the word out. And last but not least, thank you to the venue staff, Ms. Farah Mollahan, Mr. Whitney Anderson, and Mr. Josh Broussard for their hospitality and assistance in making this event successful every year.
So I want to thank you all for attending. If you like this, great. Tell your friends, please. We will continue to host this about annually, and we want as many people from all walks of the community to attend. We don't want to talk to each other. This is designed for the community. So please continue to attend and spread the word. There are evaluations. Please take a few minutes to do the evaluation, because that's how we make this better. And we do invite speakers based on the topics that you say you want to hear about. So it's really important to do the evaluations. Another thank you to the City of Houston folks who handled basically all the logistics. I'm sitting here and talking a lot. I invited the speakers. I kind of worked with them to figure out who knows what and who does what in the community. But really, Camden and his team did all the logistics behind the scene. It's a lot of work to put something like this on. And so thank you again to them. And thank you for attending. And hopefully we'll see you in about a year. Enjoy the evening.